my dear children. Let me just check if at all I am audible as well as visible. Jinchik, jinchik. Okay. Doom, doom, tada. Okay. So, hello Prajwal, hello Sagar, hello. And Pradhani and Netravati, hello Venkatesh, Sagar, and I see so many children were waiting for me. Kavana, okay, Hina, perfect. So I hope you are having a good day today. Is it a good day? All good? Hi me, okay. So hi Mallu Pawar, Swati Police Patil, hello. Tanisha, good evening. We are ready, Machi, is it? So you are ready, and if you're ready, make sure you share this video with all of your friends whom you love, because you want them also to score good. Definitely share with them as well. Yeah, Lakshmi, hello. Lakshmi, Kavita, everyone. Perfect. When will be the result announced? Which result? Are you talking about second PUC results? So it is assumed to be coming by the second week of April. Not giving you a date because I know you will be telling that JJ ma'am told and then the results have not come yet. VP Gaming, hello. Okay, so my dear children, I think we are good to go with today's live, and sooner or later all my other children will also be joining up, right? So if you had a bad day, put everything beside it, and let's have what do you call, just like how you have a new phase in your life, no, similarly a new part of your day, make it a fresh. We'll have a fun class, we'll learn a lot of things, and you will be having yeah, fun. That's all. Okay, let's go ahead then. The first thing, are we ready, Macha? I hope we are, and the list goes like this. Listen to me very carefully. We have already completed some very important chapters. Basically, we're doing PU2 as of now, right? So, sexual reproduction, human reproduction, reproductive health will be seen in class 1. So, if at all you are waiting for those chapters, I have already completed that. Go check the live, the first live of biology, okay? Now, in the second class, I did principles of inheritance and molecular base of inheritance. In the third one, I did human health and diseases and also evolution. So, if you have missed any of these classes, you have to go and check it out by yourself. Yeah? Yes. Now, going ahead with today's session, that is about biotechnology. So, biotechnology, principles and processes, biotechnology and its application. Ma'am, is it important? I always tell you, all the chapters of second PUC are equally important. Consider them to be important. Yes? Sagar, hello. Okay? So, guys, please do not spam. Okay? And over here, why I say that they are important is that if at all you look at this particular numbers. So biotechnology principle and processes, it usually gave 3 number of questions. In 2020 it came down to 2 questions, but again 3, and 1 year it went to even 4 questions. So do you think it is predictable over here? Definitely not. And from past 2 years it has been constantly giving you 3 number of questions. Okay, now biotechnology and its application also the same goes over here. It was 2, then it went up to 4, then it went up to 3, and 1 year you didn't have any questions from that, and then 3 is what the constant number has been. So obviously we have to go ahead and dig deep more into these chapters, and we will be doing that. Baslingappa, hello Kit Kat ma'am, hello dear. Deepthi, hello, and Tanisha ma'am, PYQs are enough for KCET. Dear, PYQs are not the only thing for KCET. You have to go ahead and solve other questions, but solving PYQs is important as well. By that I mean that you will get to understand the repeated concepts. While you solve the questions, you will face those repeated concepts, and while you are doing the questions again and again, don't you think you are becoming a pro at it? Yes. Right, that is why it is said that it is important. Shin-chan, namaste dear, namaste. Okay, so SSY, hello, Pooja, hello, Arjun, hello Pooja, yeah. What am I then? Okay, so biotechnology principle and processes, the first question goes like this. Which one of the following statement is wrong with respect to separation of DNA fragments on gel electrophoresis? You have to give me your answers like this. Please do not give me A, B, C, D. 1A, 1B, whatever is your answer, you can tell me like that. Gautam Buddha, hello dear. Sugamano ma'am, Amma, Malayalam, yes Sugamana, thank you. Chaya kudicho, oh my god. Okay guys, come on. C. Sagar Chopra says the answer for this question is C. What about others? What about others? B. GS World is there, hello dear, says B, and Kavita says 1D. Thank you for giving me the answer like that. Guys, please have a habit of putting the number also. Okay. Okay, B, And Pradhani says it is D. So I see C and B and D. Someone else out there with different answer. Mallu Pawar says it is B. Venkatesh says it is B. And good afternoon, Mir Jafar. Okay. Swami says it is D. Baslingappa says it is D. I see most of you guys are saying the answer for this question is D. What is there in D? Let's check it out. And by the way, Hassan, hello, hello, hello. Mohammed Javed is also there. Suresh is also there. Most of you are saying D. Listen to me carefully. You are being asked which one is wrong. First of all, when you get questions like this, underline what you are being asked. You have to find what is wrong in respect to what separation of DNA fragments on gel electrophoresis. The DNA fragments moves towards the anode under electrical field through the matrix. What, ma'am? So, Macha, listen to me out carefully. When you talk about gel electrophoresis, it goes like this. You have wells over here. Okay, you have wells over here. This is going to be your cathode. Cathode is what? It is going to be negatively charged. It is negatively charged thing. Okay. Now, here you have anode. Anode is what? It is positively charged thingy. Now, when you're putting your DNA over here into these wells, please understand your DNA is also negatively charged. Negative things it is going to repel. Opposites attract, negative they repel. Okay, the same thing they are going to repel. So over here these guys will be moving. Since it is repelling, obviously it will be moving away from it. Away and towards what? Towards the anode. So the DNA fragments move towards the anode under electrical field through the matrix. The answer would be correct. This is particularly correct, right? It is moving towards the anode only. Now the commonly used matrix is agarose gel. So over here the gel that you're going to use is what? Agarose. Is it agarose gel? Yes. Where do you get agarose gel from? From the seaweed out there. Okay, it is going to be from the seaweed. Okay. Now this is also correct. Now C option is the DNA fragments resolve according to their size. Do they move according to their size? What do you think? Do they move according to the size? Yes. Listen to me carefully. Now the bigger one which is there and the smaller one which is there, which one will be able to move the fastest? It will be the smaller one. Why? Because you have to use your imagination. The gel is right over here. It's not like the DNA is moving on the gel. It is moving within the gel. Now within the gel, if some chutku guy is there, it will be able to move around, right? But if at all it is big, it is going to face inhibition. It is going to face inhibition, which is why the smaller one is always the one that moves the fastest and also the farthest. That means it will be moving very far from the starting point out there. So DNA fragments resolve according to the size. This is also correct. Now statement number D says the smaller DNA fragments separate first. Ma'am, you only said that this particular thing is correct. Right now you told that smaller fragments move fastest. I did tell that the smaller fragments move fast. I didn't tell that they separate first. Listen, from this particular area, it can be the larger one that separates first or the smaller ones. How can I know that? No one can tell that. If at all you're talking about who moves the fastest, I can tell. Who separated first, I cannot tell that. So this particular statement is going to be wrong, which is why all my dear Chitko champions who gave me the answer as D, you're absolutely correct. Okay? Correct. So D is the answer. So most of you, I see Mallu Pawar, Chris is there saying the answer is D. Mir Jafar also says it is D. Deepti also says it is D. Suresh D. Mohammed Javed D. Baslingappa says D. Swami says D. Venkatesh says D. Okay. Kavita also said D. That is what I got. Now, my God, just a second. Guys, so many questions. Ma'am, you told solve each question in 1 minute. How in starting it's not easy, then how to manage PU2? Sagar, in starting it will not be easy, but you keep the timer, you start solving, it automatically will be coming in the flow. It will be coming in the flow. Okay? Mir Jafar, ma'am, in your class we get interest to learn and we interact with each other. Thank you for being so close to us. Thank you so much for telling that. And hello, ma'am, in KCET ABHYAS, this test series, the questions will be new. Okay, they are marked as they are different type of questions which will be found up there. Guys, if this particular concept or question is clear to you, can everyone give me a thumbs up? This is the rule. After every question, you have to give me thumbs up. That's when I understand that you children are understanding things. Okay? Words. Sagar is not understanding. Ma'am, it is not clearly visible. Guys, is it not clearly visible? Children, is it clearly visible or not? The screen is visible. It's visible. So I request you to please increase the brightness, dear, if at all someone was facing difficulty. Ma'am, what to do to score 100 plus in KCET? Know the concepts, solve the questions related to the same concepts, and practice mock tests. That's the only way out there. There is no shortcut up there. Okay, perfect. Thank you. So let's go ahead with the next thing. Question number 2. Elution means what? Separation of DNA fragments on agarose gel. B. Cutting and extraction of DNA bands from the agarose gel. Making the DNA bands visible under the UV radiation. Isolating alien DNA from the choice organism. What do you mean by elution? Your answer goes like this. To A, B, C, D, you have to tell me, okay? Shamant is there. Hello, Shamant says the answer is A. Mirzafir says C. Okay. Hassan says it is A. Deepti says it is B. Anant is there. Hello, hello. Says A. Swami says Macha, B, Macha. Okay, B. Okay. Hina Kausa says B. Sagar says B. Purvi says B. Suchit is saying B. Tashrif says A. Crystal Fernandez says B. Venkatesh is also saying B. And Sinchan has a different answer. Okay. Prajwal says A. Okay. So most of you guys are saying B. And I see confusion between A and B, which is why I need to solve it. Listen, over here, you separated the DNA fragments. Suppose over here you separated DNA fragments. These DNA fragments is within the gel. You have to take it out. You have to remove it. You have to cut and extract it. This process is known as elution. Why is this A not correct? Separation of DNA fragments on agarose gel is not elution. You're not separating. You're cutting and extracting the DNA bands from the agarose gel. That is what you mean by elution. Making the DNA bands visible under UV radiation. No. How can you make the band visible as of now without using a stain? Isolating alien DNA from the choice organism, that is also not correct. So which is why your answer for this question is going to be what? B. It's a simple question. Okay, but I definitely get it. If at all someone has missed by any chance the term elution, you might not know it. But you know it now, right? Arif, good evening, dear. Ma'am, by mistake I put it. It's okay. Perfect. Clear? If it's clear, just give me a quick thumbs up. Thank you, Shamant and Hassan and everyone. Sorry, ma'am, I missed the question. It's totally okay, Pooja. That's how we learn, right? Okay. Good evening, Sanika. Okay. Perfect. Thank you so much. Just a second, children. I will take a question. Ma'am, in KCET ABHYAS, the questions are really blurred. Whom should I contact? Please answer me. The questions are blurred. I'll raise the concern if at all it is there. Okay, Shreyas? Give me some time, today's time, okay, I'll just raise the concern. We didn't get any of these complaints before, so I'm like a bit skeptical. Yes. Yes, and let's go ahead with the next question that is up there. Restriction endonucleases are isolated from some bacteria. Their role in bacteria is what? Restriction endonucleases are isolated from some bacteria. What is their role in bacteria? What are they doing there, Macha? Defense against virus, synthesis of proteins, act as genetic material, help in reproduction. What do you think is the answer for this? Pradhani says C. Okay. Shaman says B. Okay. Deepti says I do not know, dear. Okay. Suchit says A. Deepti says A. Purvika says A. Sorry, Purvi says A. Aditya, Crystal have, okay. Sanika, a lot of you have different answers. And most of you are saying C is the answer for this question. What is C? Acts as genetic material. Guys, listen to me very carefully. Restriction endonucleases are isolated from some bacteria. This is the bacteria, suppose. What is this bacteria? Let's name this bacteria Chutku. Okay, this is Chutku bacteria out there. Actually, the name is E. coli. Okay, E. coli will be having its own genetic material. Do you disagree? No, you should not be. Okay, it has its own genetic material. Now, suppose one virus came into the picture. This particular virus, what does it do? It puts down its genetic material inside it. Ma'am, why? Because it will be slowly trying to incorporate over here, use the cell's machinery to basically produce its own viral components out there. You might have seen HIV virus, how does it work, right? Same thing is going to happen over here. But our Chutku Macha, he is very clever. He said that you cannot do that because I am going to produce two things over here. One is restriction endonuclease, which is the enzyme, and the other enzyme that we have over here is something that adds methyl group. Something that adds methyl group. Okay, now the story goes like this. Over here, Chutku releases restriction endonucleases to cleave off or to cut off the genetic material of the organism, the foreign organism's genetic material out there. Okay. Now, when it produces restriction endonucleases, you need to understand that this enzyme can cut its own genetic material as well. right? So to be protected from this particular enzyme, with the next enzyme that is there, it is going to add some methyl group up over here. So when methyl group is there, the enzyme will not act on it. The enzyme is not going to act on it. Here the question is, what is the role of restriction endonucleases in the bacteria? That is the question. So it will be what? Defense against virus. It will be defense against any foreign thing, but particularly the virus. Synthesis of proteins? No. Act as genetic material? They have their own genetic material. Help in reproduction? No. Your only answer for this question is going to be A. Is this particular thing clear? Ma'am, make it Pappu, not Chutku. Oh my God. You guys remember everything. Yes? Is this particular question clear to you? If yes, give me a thumbs up. So all those of you said A was the correct answer. From the starting point onwards, kudos to you. I might not be able to read out your name, but it means a lot. Okay, you guys are working really hard, and those of you who got it wrong, it's okay. Chill. Okay, you now know the answer. Do not repeat the mistake. That's all. Fine. Thank you so much. Most of you have understood. Let's go ahead with the next question. Question number 4. From which bacterium the REN, that is restriction endonuclease, SAL1 is isolated? Okay, SAL1. From which bacterium do you get? Ma'am, out of the syllabus question. Is it? Ma'am, these bacteria, I just know Escherichia coli, Streptococcus aureus and all, I do not know. Haemophilus influenzae, I know. Streptococcus albus also, I do. Streptomyces albus also, I do not know. Out of the zone question. What do you think is the answer? Just chill. Okay. So I think the first person to tell the answer is Pradhani says A. Okay. Kavita says the answer for this question is D. Hassan says A. Suchit says D. Mallu Pawar says A. Purvi says D. Sagar says D. Shaman says A. And Swami says D. Macha. Okay. Deepthi also says D. Shrira is there saying A. Anand says A. Arif says B. Okay. Hina Kaushal says A. Which means that we are a bit confused. And most of you guys, Lavender is there, Tanisha is there, and Tashrif is there. Okay. Okay. Anusriyano is also there. Venkatesh is also there. Listen to me very carefully. The answer for this question is not A. Here, the logic behind this particular thing is we know restriction endonucleases. No, when we talk about restriction endonucleases, you have studied. For example, EcoR1 is a restriction endonuclease. What does E stand for? E stands for the first letter of the genus name, Escherichia, E. CO stands for what? The second 2 letters of the species name. The second 2 letters over here is basically the first 2 letters of the species name. That is coli. Coli. These 2 letters I have taken over here. Right. Now what does R stand for? R stands for the strain, whatever strain it belongs to. And I is the number of discovery. In which position, basically in which order was it discovered. Now when you are saying you are given this particular thing, that is the name of this particular enzyme is SAL1. S will be basically what? S will be the first letter of the genus name. Over here I have 2 options only with the first letter as S in the genus name. One is Streptococcus aureus and one is Streptomyces albus. Right? Yes. So I just have 2 options. Now talking about AL, the second 2 letters will be what? Of the species name. The first 2 letters of the species name are the second 2 letters over here. So over here you see AU. It is AL. Here you see AL. So this is going to be the answer. The answer for this question is going to be D. Okay, it is going to be D. Is it clear? Is it clear to you? Streptomyces albus, this is the answer for this question. Very simple. Okay, now you might think that, ma'am, for you everything will be simple. No, I'm also like you. Trust me on that. Okay. Sorry, ma'am, I got confused. It's totally fine, Macha. Okay, perfect. Let's go ahead then with the next question. Yes. By the way, everyone who's watching this video, have you hit on the subscribe button? If not, hit on the subscribe button right now and hit on the like button as well. When you click on the like button, what happens? It basically goes to all the children out there who are in the zone who wants to study for KCET. That's how it goes. Okay? Yes. Okay, Sagar, I got correct. Perfect. Sandy, hi Machi. Hello dear. Hello Sandy. Hello Sandy. And Prajwal, oh my God. No dear, it's a wrong notion. Okay, let's go. A transformed bacterium with human gene fails to produce desired protein. The reason could be what? A transformed bacterium with human gene. Basically, this particular bacterium which is there, it has a human gene. This is what? A gene of human being. Okay. and it fails to produce desired protein. This particular gene is not able to make the protein out there. Why do you think that is happening? Human gene may have intron which bacteria cannot process. Amino acid codons for codons for human and bacteria are different. Human protein is formed but degraded by bacteria. The bacteria promoter region cannot induce transcription of human gene. What do you think is the answer for this question? A. Oh my God. Everyone says, most of you guys says A is the answer for this. A is what? A is human gene may have intron. Exactly. You guys are awesome. So listen to me very carefully. The DNA of human genome will be containing introns that do not code for anything and also exons that code for the amino acids out there. Usually what happens is that when we do transcription, DNA, it transcribes into hnRNA. The same thing happens over here. I and E portion, everything will be there. Now, when you form the mature RNA, which is finally going to make the protein, you will not have these guys up over here. Introns are going to be removed. You should be only having exons. You will be only having exons. In the case of bacteria, they never have introns. So they do not know the mechanism of how to remove the introns. But we have it. So when we put the DNA inside it, basically what happens, the introns also goes along. And the gene will not be able to express itself. Why? Because the intron portion are not removed over here. So which is why your answer for this question is going to be A. It is going to be A. Amino acids of codons of human and bacteria are not different. You know, amino acid codons are universal, except some exceptions are there, right? Human protein is formed but degraded by bacteria. Why will that macha do that? No. The bacterial promoter gene cannot induce. That does not make any sense. So over here, this is the answer. Yes, exactly. Okay, perfect, works. If it's clear, just give me a thumbs up. Tasmi, I love you. What is happening over here? Guys, this is YouTube and this is Jasmine ma'am's class, biology class. This is not Tinder. Come on guys, over here, everyone. Make sure you listen to me carefully. Ma'am, I'll start with, okay, perfect. Sagar, wishing you all the best. Ma'am, in KCET we should solve everything and we need to tick a, you need to, in KCET you need to fill the OMR. Yes. Hello ma'am, so sorry for late. I found you yesterday, so I decided class I attend from tomorrow. Okay, you're here finally. Okay, let's go ahead then. Next one, question number 6. Identify the palindromic sequence in the following base sequences. Okay, you have to find what? The palindromic sequences. Come on guys, go ahead and find the palindromic sequence. Go ahead and study the palindromic sequence. Ma'am, make video on OMR. Yes, I will be doing that. Okay, so most of you, Sandy says D is the answer, Tasmia says D, Pradhani says D, Suchit says B is the answer, Kavana says B, Swami says Macha B, okay. Tashrif says D, most of you guys I see it's B. Shinchan and Ansuya and can I call you Anu? I hope you do not mind. Purvi and everyone out there. Most of you are saying the answer for this question is B. Monish, Noor the Kitty, everyone. B is what? B is this. What do you mean by palindromic sequence? The one that is read in the same way from both the ends. For example, you know the language Malayalam? Malayalam. Okay, when you talk about Malayalam, it is read in the same way from here, the same way from here. So over here, you have to find the palindromic sequence. So let's check it out. 5-CGATA. Okay, so from here, when you read it out, do you think it is C? No, it is not C, it is T, wrong. Okay, GGATTC, 5- to 3-. Now, ulta. 5- to 3-, GGATTC. It's the same thing, it is read in the same manner, so this will be your answer. But sometimes we get confused, so we have to check the other options as well. CCTGG, okay. Then from here, GC, obviously wrong. Next option that we have is GAATTG. From here, if at all you read, it is CAA. So obviously it is also going to be wrong. Your only answer for this question is going to be B. It is going to be B. Okay, perfect, works. Isn't it necessary for it to be GAATCC? Huh? Yes, it should be. It should be the same on both the sides. It should be read the same in both the ways. And always remember, they can give you some ways. It's okay, some question where they have mentioned as this is also 5 dash to 3 dash. Make sure you check that as well. You have to check that as well. If this is in 5 dash to 3 dash, the other one should also be in 5 dash to 3 dash. They will give you the exact sequence. This will be correct. This portion will be correct. But they might be making you confused with the tagging out there. Okay, the polarity out there. Don't get confused. Okay, works. Let's go ahead then. Okay, why not D? Why not D? D, C over here. G-A-A-T-T-G is there. From here also it should be read like that. G-A-A-T-T-G. But here it is C-A-A-T-T-C. That is why it is not D. Cool. Works. Okay, let's go ahead with the next question out there. Question number 7. The technique of bombarding the plant cells with high velocity microparticles of gold and tungsten coated with DNAs. And my dear children, this particular question is important. I'll tell you, you have get so many questions on the basis of this particular concept. So many questions. I think 5-6 questions were there. Okay? Okay. B again. Sandy says it is A. Kavita says it is B. Okay. Suchit says B. Tasmiya, Kavana, Hassan. Okay. I'm seeing your answers. Hina, Deepti, Shrira, Tasmiya, Swami, Purvi, Sandy, Sannidhi, Ranjit, GS Suni, Tasmiya, Lavender, Pooja. Okay. Lilavati, Anu, okay. Suchit, Sarveen, Pradhani, Tanisha. Okay. Listen to me carefully. Arif. Over here, the answer for this question is going to be B. It is going to be B. Okay, so most of you have given me the answer as B, but some of you had given some other answer. Listen, it's very simple thing. This comes under what? This comes under what do you call vectorless methods of transfer. Okay, this comes under vectorless methods of transfer of recombinant DNA. Suppose you have a plant cell. This plant cell, Baba, will be having what? An extra layer over here. That is known as cell wall. It is known as cell wall. This is what? A plant cell. Now, on the other hand, contrary, you have to think about this also. There is one more cell over here. This is basically an animal cell. This is an animal cell. Does it have a cell wall? No cell wall. Does it have a nucleus? Obviously yes. This guy also have a nucleus? Obviously yes. Now I want to transfer the DNA, the recombinant DNA from outside without using any vectors. I cannot use any organisms in between. What can I do? In the case of animal cell, I'll just take a normal sort of injection sort of thing and I am going to inject my recombinant DNA. This is known as micro injection. Micro injection. But when I try to do the same thing over here, what is going to happen? I'm trying to inject, but ta-dang, what happens? The needle breaks off. Why? Because you have a cell wall over here. Obviously, you cannot do that. Micro injection is not going to happen in the case of plant cell. Then what do we have? We bring up a gun. Okay, we bring up a gun and we are going to use, instead of bullets, what do we have? We are going to have microparticles of gold and tungsten. Gold or tungsten microparticles will be there. We are going to surround it or coat it up with the recombinant DNA and then ta-dang, we are going to put it inside. Okay, we will be putting it inside. This is known as gene gun method. This is known as gene gun method. But there is other name which you always forget sometimes. Okay, not always, sometimes you might forget it, and that is biolistic method. The same thing is known as biolistic method. And that is the question over here. When you're doing this, when you're using microparticles of gold and tungsten and you're putting inside, what is this? Micro injection, biolistic method, heat shock method, or by disarmed pathogen vector? The answer is going to be biolistic method. And this is the concept that I was talking about. So many questions have come from the same concept about biolistic method. So make sure you are a pro at it. Okay? Is it clear? Ma'am, I'm not boy, I'm girl. Oh, sorry, dear. Okay. Ma'am, you're cool and chill, so we love you. Thank you so much. Okay. Biology is easy for NEET, but not for KCET. Is that so? Never. It's easy. You will be pro at it, then you will have it, what do you call? It will be easy for you. Cool? Ma'am, I have a doubt regarding passing package of science theory paper. Passing marks. Okay, sorry. Is it 21 or 24? I have to check. Christopher, since I'm not in the answer checking group, okay, I'll check it and I'll let you know, okay? If you can, after the live ends, put it down in the comment section. I can reply to that. I'll remember it then, okay? Hi, Sanket. Good evening. And Maunesh, guys, do not talk to each other over here. Adigittu. Get off. Okay, Mir Jafar, ni valare sundriyanu, valare elegant and oh my god. Thank you. Okay, guys, let's go ahead with that ChatGPT Malayalam. Exactly. So anyway, let's go ahead. Let's go ahead with the next question. If it's okay for you, if you have understood this particular concept, just give me a thumbs up, everyone. Everyone, just give me a thumbs up. So how many questions to complete in one class? So over here, I always target 50 questions mostly. So today also 50 questions are there. Hassan, thank you, Da, and Sandy, Sai, and Ranjit. Okay, perfect. Arif, thank you so much. Let's go ahead with the next question out there. Question number 8. To isolate DNA from the fungal cells, be very careful over here. To isolate DNA from the fungal cell, bacterial cell, and plant cell, the enzymes required are respectively what? What are the enzymes that you require over here if at all you want to isolate the DNA from either of these cells out there? Yes, ma'am, sorry. It's okay, Monish. I forgive you. Okay. C, Suchit, Ganesh, Nayak, Hassan, and Kavana, Purvi, Tasmia, Dadipti. Oh my god, so guys are so fast. Arif, GS Sunny. Okay, Sannidhi, Srirar, everyone, Hina Kausar, Servine, Tasmia, Rohit, Shin. Okay, Okay. So, Manjula, everyone says the answer. Sai also says, Noor the Kitty also says it is C. What is C? Chitinase, lysozyme, and cellulase. For those of you who did not understand, listen to me out carefully. Suppose this is a fungal cell, okay, and this is, I'm writing F. And over here, this is a bacterial cell, and this is going to be what? A plant cell. Guys, you all know that these cells which are there, they have an extra wall layer, which is known as cell wall. All of them have a cell wall. But the components which if their cell wall is made, it is different. In the case of fungus, the cell wall is made up of chitin. Okay. In the case of bacteria, the cell wall is made up of peptidoglycan. Okay. In the case of plants, the cell wall is made up of what? Cellulose. Now, I am targeting for the DNA which is inside. Obviously, I have to break the cell wall open first, right? Which is why, if at all I want to isolate the DNA over here from a fungal cell, I am going to require 3 enzymes. Over here for the fungal cell, I require chitinase, that is going to break down the chitin. For breaking down peptidoglycan, you have lysozyme. I know the name and this does not sound similar, but yes, it is. Lysozyme is the enzyme that you need to break down, that is used for breaking down of the bacterial cell wall. Now, cellulase is there. Cellulase for breaking down of cellulosic cell wall in the case of plant cell, which is why the answer for this question is going to be C. Now, what type of questions can be coming off from this is that they can give you the name of a fungus, okay, and they can give you a name of a bacteria. If at all they're asking you from an E. coli, if at all I want to isolate the DNA, what enzyme will you use? So how will you tell it? Basically, if it is E. coli, it's a bacteria, then it will be lysozyme. And do you know that in our tears, when we cry, we say that sometimes crying is good. Why? Because in our tears also there is lysozyme. So when something goes into our eyes, we'll be like, oh my god, I'm crying. There's a lot of water coming out, right? It's a defense mechanism. Your tears have lysozyme, and the lysozyme kills the bacteria up there, if at all there are any. So everyone who gave me the answer, you're absolutely correct, and I'm super proud of you. If you have understood the concept, give me a thumbs up. Everyone who have understood the concept, are super clear with a lot of things, just give me a thumbs up. Saline water, exactly. From tears, I remember lacrimal gland. Oh my god, yes. Okay. Oh my god, yes. It's fake, Prajwal. Okay. Yes. So let's go ahead with the next question out there. Question number 9. The parts of plasmid that codes for proteins involved in the replication of PBR322 plasmid is. The part of plasmid, or basically the sequence of the plasmid or the region of the plasmid that codes for proteins, that is very important for the replication of PBR322 plasmid is what? What do you think is the answer for this question? C. Sandy says it is C. Okay, Suchit says it is C. Sandy's changed the answer. A. Okay. You're most welcome, Prajwal. Okay, Kavana says C. Savita says C. Monesh says C. Deepti says C. Noor the Kitty C. Malu Pawar, Tanisha, Kavana, Puri, Hassan, Deepti. Okay. Most of you guys are saying C. Okay. Ma'am, I see your previous section for 50 questions. Your class duration is much high, but good for understanding each and every question. Thank you so much. Yes. Kavita, ROP, it was there in second PUC also. Oh my God. Yes. All of you who have said the answer for this question is ROP, it is correct. ROP. For those of you who do not understand it, listen to me carefully. Okay. We'll have a fast fast revision of this. Fast revision of PBR 322. Listen, your PBR 322 looks like this. You have an origin of replication site over here. This site is required for the whole replication to happen, and also it decides the copy number. How many cycles of replication can happen is decided by ORI site. Then over here you have an ROP site. ROP site is basically a site that is going to code for proteins. You know, proteins are also required for replication process. The proteins for replication are made by this particular site, and that was the exact question over here, right? Now you have two major sites over here. This gene over here is going to help in providing resistance against ampicillin. Ampicillin is an antibiotic, okay? Suppose an organism has this plasmid, and this plasmid, you know, it has a gene to resist ampicillin. So that means that that organism can live in a medium which has ampicillin, that is an antibiotic. It can live, why? Because it has a resistant gene out there. Similarly, there is another gene, tetracycline resistant gene. This is another enzyme, antibiotic, sorry, not enzyme, antibiotic resistant gene out there. Now, there are multiple sites, multiple restriction enzyme sites over here. What are the restriction enzyme sites over here? Let's go ahead. So always remember, one pista shake, so pista 1, one vada pav, PVU 1, okay, pista shake 1, PVU 1, done. These are the sites over there. Now here you have ecology class, eco R1, class, clav 1, and Hindi class is also there. Third period is Hindi, Hind 3, okay. Now in tetracycline, Baba, what do you have? Tetracycline gene, here also you have site. One is BAMH1. Oh my god, I BAMmed into Salman. So BAMH1 and Sal1, okay. And finally in the ROP site you have PVU. You know, vada pav 1 was there, here you have 2 vada pav. Okay, so pista shake 1, PVU, and I made up a story when I was talking, when I was teaching where? In second PUC, and that story was literally very bad. But if at all you need to hear the story, I can tell right now also. So listen to me carefully. I know you do not want to, but listen, this might help you. So two children were there, okay. One had pista shake, one had one vada pav, okay. They had actually, they went to, they were ecology students, okay. They went to Hindi class, Hindi was the third period, and they BAMmed into Salman. And they took Salman also, and they had one more vada pav. So it is 2 vada pav now. VV2. Okay. I am also remember like this only. Yes. So yes. So that is the thing. Works. Clear? So I know that does not make any sense, but yes. Sometimes silliest of the things can help you remember things when in need. Okay. Ma'am is flexing her phone. Where is my phone? I have not shown my phone, only chumma. Okay. Clear, right? Give me thumbs up. Bemisal. Oh, ecology class Hindi teacher Bemisal. Oh, nice. Yeah, but anyways. Okay, guys, let's go to the next class. Next class out. Oh my god. Next question out there, question number 10. If a recombinant, okay, this is important. This might confuse you, so read the question carefully, okay? If a recombinant DNA bearing gene for resistance to ampicillin is transferred into E. coli cells, once more, if a recombinant DNA bearing gene for resistance to ampicillin is transferred into E. coli cell, okay, host cell becomes transformed into ampicillin resistant cells. What happens when these E. coli are grown on medium containing ampicillin? What happens when these E. coli are grown on medium containing ampicillin? A. Non-transformants will grow and transformants will die. Okay. B. Non-transformants will die and transformants will grow. Both non-transformants and transformants will grow. Both non-transformants and transformants will die. Where is it will grow? Okay. Okay, I think it is, okay, will die is the option over here. Yes. What do you think is the answer for this? Tanisha says the answer for this is B. Okay. Deepthi says it is A. Swami says it is C. Okay. Arif says it is C. Pavitra says it is B. Sandy says B. Swami says C. Guys. Be very concentrate really well, okay? This question is important for you, and I've seen multiple questions coming from the topic. B exactly. Kavita says B exactly, okay? Kavana says B. Prajwal says, I think D. Anushya says, GS says B. Most of you guys, Pooja is also saying B. Pavitra B. Okay, most of you guys are saying B, which is why, ma'am, some confusion is there. Yes, I know some confusion is there. Sagar, we will be doing that Okay, don't spam, dear. Okay, listen to me carefully. Why do you think it's D? Why do you think it is D? It is going to be B. Why B? Over here, I have, suppose, this is very important concept, okay? These are 2 E. coli, or basically I can say these are 3 E. coli. Okay? And this is your plasmid that is having ampicillin resistance, right? Yes. And this is going to be another plasmid. If a recombinant DNA bearing gene for resistance to ampicillin is transferred into E. coli host cell, becomes transformed into ampicillin. Okay, fine. So basically, I have a scenario which goes like this. If E. coli 1, which is there, it does not take any plasmid from outside, do you think it will be resistance to ampicillin? No. So I'll write no resistance to ampicillin because it have not got the gene. No resistance to ampicillin. Okay. If the E. coli gets this particular plasmid which carries our ampicillin resistant gene over there, do you think this is E. coli number 2? Okay. Do you think it will be resistant? Yes. It is resistant. Now, one very important question before I go ahead. You know which is resistant, which is not. Which one is a transformant one? Which one is a transformant one? 1 or 2? Which one has got transformed? 1 or 2? Listen to me carefully. If you did not have anything, you have changed, okay? You say this is transformation that is being seen, right? Similarly, the E. coli platelet didn't have anything. It took the recombinant one. That one is going to be the transformant. Okay, the one that did not take anything is going to be what? A non-transformant. So 1 is going to be non-transformant, 2 is going to be the transformant. Now give the answer. Non-transformants will grow and transformants will die. How can non-transformant grow? Because you are growing this E. coli in an ampicillin medium. If you have the superpower to grow even when ampicillin is there, then that's different. But over here you do not have that superpower. You have not got the plasmid. So this statement is going to be wrong. Non-transformant will not be growing. It will be dying. Non-transformants will die and transformants will grow. Yes, it is going to grow. Why? Because it is resistant. Transformant will be living. Non-transformant is not going to live. Both non-transformant and transformant will grow. That is obviously not possible. So which is why your answer for this question is going to be B. Is this particular thing clear? Yes. Yes, Sanket, Anusya, as well as Prajwal. Is this particular thing clear? Any idea, guys? Any idea until what time the class is there? It will be mostly classes go up to 8 o'clock, but I'll try to finish fast. But mostly it goes, okay? Yes, perfect. Give me a thumbs up if this is clear. And one more thing, this is transformant. If you're asked what is recombinant, how do you answer that? Transformant is different, recombinant is different. Listen. One more scenario I am going to give you. Do not worry, I will not explain much. This is for you guys only, not for me, right? Am I going to write KCET examination? Definitely not. So, here, I am saying that this particular thing is ampicillin resistant gene is already there. And it also has my gene of interest. My gene of interest is also there. I have not just received a plasmid, a random plasmid. I have got a plasmid that has my gene of interest. So I will call this as a recombinant. It is transformant because it has got a plasmid. It is recombinant also because it has got a plasmid with gene of interest. Whereas when you look over here, it has just got a plasmid. So it has transformed, but it has not got the gene of interest. So it will be non-recombinant. So please know that particular concept as well. Okay? Yes. Let's go. Now, question number 11. DNA polymerase of Thermus aquaticus is very simple question. Thermos, labile, thermophobic, exonuclease, thermostable. What do you think is the answer for this? Hello, Vidya. Hello, hello, hello. Okay, ma'am. Thank you so much. You're most welcome, Crystal. Who will themselves change the situation? They will not die, but who will change? Yes, exactly, Sagar. Perfect. D. Facts is also there. D. Very easy question, right? It is D. D is what? D is thermostable. Yes. DNA polymerase of Thermus aquaticus. This particular organism can stay at high temperature. And basically, we are taking the DNA of that particular organism. Why? Because you know there's a process, polymerase chain reaction. And in that process, you are going to replicate the DNA at high temperature. So you need an enzyme for doing so. And that is DNA polymerase. And that DNA polymerase which you take from here, you also call it as Taq polymerase. First letter from here and second two letters from here. Cool? Yes. So it is thermostable. Perfect. Let's go ahead with the next question out there. Which of these is not a method to make host cells competent to take up the DNA? Which of these is not a method to make the host cells competent to take up the DNA? Elution, biolistics, use of disarmed pathogen vectors, microinjection. Not a method to make a cell competent means the ability to take up the DNA. That is what you mean by competent. So you are asked, what is that method with the help of which you cannot make a cell take up a new DNA out there? A. Most of you are saying A is the answer. Okay. So from which point onwards have you started giving me the answer for this question? That I'm not able to understand. Okay. A, Baba, absolutely A. Okay, Baba, absolutely A is going to be elution. We studied elution is what? The extraction, cutting and extraction of DNA from the gel electrophorese chamber. So which is why the answer would be A. Done. Next question. Select the correct statement from the following. Guys, for this question, can you give me the answer like this? 13 ABCD, because I'm losing the track of it. Okay, 13 ABCD. That will be really good. Okay, select the correct statement from the following. Correct is being asked. There are no risk factors associated with our DNA technology, that is recombinant DNA technology. Okay, the first step in PCR is heating, which is used to separate both the strands of gene of interest. In PCR, the first step is what? Is it heating where you're going to separate the strands out there? Think about it. Okay, see, DNA from one organism will not bind to the DNA from other organism. Is that correct or wrong? Okay. D. Genetic engineering works only on animals and not yet successfully used on plants. Is that correct or not? You have been asked which particular option is correct. Okay, which option is correct? And I see. So those of you started saying 13. Okay. Sanket says 13B. Hello Sham Kamle. Okay. Deepthi says 13B. Shivanand says B. Sagar says B. Darshan says D. GS Suni B. Arif, Tasmiya, Rekha and Swami and Vidya, Sanket, Suchit, GS World, Pooja Pujari, Mallu Pawar. Okay. Hassan. Perfect. Sandy, everyone. Crystal Fernandez, Hina Kausar, Vidya, Sinchan. Perfect. The answer for this particular question is going to be B. It is going to be B. So there are no risk factors. Obviously, that's wrong. There are definitely risk factors which are involved with... Sorry. What is the correct statement is being asked? So there are no risk factors would be wrong statement, right? There are definitely some risk factors out there, which is why this is going to be wrong. Okay. Now, the first step in PCR is known as denaturation, where you are going to separate the DNA using high temperature. The temperature is almost around 90 to 95 degrees Celsius. So this is also correct. This is the only one which is going to be correct. DNA from one organism will not bind. It will not bind. That is pretty much clear. It will not bind with the DNA of other organisms. We know that. So this is also correct. Sorry. Yeah, genetic engineering work, just a second, I am getting confused, just a second. The answer for this particular question is going to be B, and you have been asked what is the correct statement. So, this is correct, this is going to be correct statement. There are, okay, just a second. Guys, my brain is not braining. Select the correct statement from the following. Okay, so there are no risk factors. Definitely there are risk factors, which is why that is not correct. Okay, DNA from one organism will not bind to the DNA of another organism. That will be wrong because it can bind if at all the complementarity is there. Genetic engineering works only on animals and not yet successfully used on plants. that will be again wrong because it has been used on plants as well, which is why the answer for this question is going to be B. Yes. So this happens when my brain is not braining. Okay. So I'm so sorry. It happens. Hiyo. Okay. B is your answer for this particular question. So for a slight second, I got zoned out. Macha, cool, solve. Okay, yes, thank you. Next question. Question number 14. From the following tools, techniques of genetic engineering, identify those which are required for cloning a bacterial gene in animal cell and choose the correct option. This is a very important question. It's okay, ma'am. Thank you for understanding. Yes. So what do you think is the answer for this? What are the things that you're going to use if you want to clone a bacterial gene in an animal cell? Remember always, just write it down. Bacteria and animal cell. And just think of what all things do you require over here. Endonuclease. Endonuclease do you require over here? Endonuclease means restriction endonuclease. You use that for cutting the genetic material out and putting it right? So this is required definitely. What about ligase? Ligase is what? Again, you have taken the genetic material, you have to insert it somewhere, and you have to make sure that it sticks also. So you need the enzyme ligase, because ligase is the one that is there for sticking things up. Okay. Agrobacterium tumefaciens. Do you require Agrobacterium tumefaciens? Listen, over here you're talking about animal. You want to put what? You want to put the bacterial gene into the animal cell. In the animal cell, you need other vector, not Agrobacterium tumefaciens. This is something that we use for inserting things into the plant, not animal. So this is the odd one out. Microinjection. Do you need microinjection? Yes, you can take the help of microinjection for animal cell. There is no cell wall, so we can do that. So this is also correct. Gene gun. Again, this is for plants, not for animals. So wrong. Lysozyme, do you require it? Lysozyme is what, ma'am, we do not require it. You require it. Why? Because you want to break open the bacterial cell wall. And the bacterial cell wall is made up of what? Peptidoglycan. So obviously you require this, right? Then cellulase. Cellulase is what? To break down cellulose. Do you have plant cell over here? No. So you do not require this. Electrophoresis. Obviously, yes. You want to separate the DNA which you have just taken up, and you will take the small strand which is useful for you. So you require that also. So what are the things that you require now? 1, 2, 4, and 6 and 8. 1, 2, 4, 6 and 8. Your answer is going to be A. So for everyone, okay. Now for everyone who said this, perfect. Cool. Darshan, Arif, and Savita, Shivanand, Tasmiya, Malupawar, Vidya, Yogita. Hello, I am Ganeshi. Hello, Ganeshi and Sandy. Dear, all my dear children, I hope this is clear. If it's clear, just give me a quick thumbs up. If it's clear, just give me a quick thumbs up. It's too hot. Oh my God. Ma'am, poll. Dear, poll is not over here. So sorry. That software is entirely different. Okay. Okay. Yes. Perfect. Thank you for giving me the thumbs up. And yes. Let's go ahead then. An antibiotic resistant gene in a vector usually helps in the selection of an antibiotic resistant gene, ampicillin resistant, tetracycline resistant, right? Remember, in a vector usually helps in the selection of what? Non-recombinant cells, competent cells, non-competent cells, transformed cell. What do you think is the answer for this question? This chapter is so confusing. I must agree to that, but not exactly. Karthik, ma'am, you're so sweet. Thank you so much, Macha. Okay, so it's okay. Where are we? Deepthi says A, Rekha says A, Savita says A, Hassan says A. Okay. Suchit says D. And Sol, hello Radhe, hello. And Kavita says D. Okay. Sanket says 15A. Shivanand, guys. Fax says D. Tanisha says D. So now I'm just reading out the children who gave me the correct answer. Okay. Lakshmi says D. Darshan says D. Answer is going to be D. An antibiotic resistant gene in a vector usually helps in the selection of transformed cells, not non-recombinant cells. Why do you think it is non-recombinant? Walaikum assalam. Assalamu alaikum. I do not know how to reply for that, but yes. Perfectly. Feeling bio easy because of you. Thank you so much. Guys, Nice. What is the answer for this particular question? It is going to be D for this particular reason. If you are not understanding this, listen to me out very carefully, okay? I told you that one particular cell which is there, suppose you have three different scenarios. You have three different scenarios. This cell has not taken up anything. Okay, that means it is not resistant to the gene. Not resistant to what? The antibiotic, not the gene. Okay, this has taken up. This has taken up, right, the ampicillin resistant gene. So it is resistant over here. This is also there. It has ampicillin resistant gene. Along with that, it has the gene of interest as well. It has the gene of interest as well. Anyway, Baba, it is going to be resistant. It is going to be resistant. Why? Because it has ampicillin. Over here, aren't both of them transformants? Aren't both of them transformants? Aren't both of them transformants? Can I tell, looking at this, that this is not recombinant, that is recombinant? No, I cannot tell that. So the first thing, when you are using antibiotic resistant gene, the first thing that you can find out is whether they have got transformed or not. So the answer for that particular question is going to be D. Is it clear? Is it clear? I know, if we solve calmly, then D. Exactly. Exactly. If we solve calmly, then D. You saw, me being a teacher, I got stuck at that particular question. Why? Because I am in a hurry over here to, okay, go ahead with the next question. A lot of things are happening and we are trying to find the answer. But on the other hand, when we sit calmly out there, we look at the question, we get the answer right. So all of you who made mistake over here, we have got a learning. Okay, we all have got a learning. Take it slow, take it calmly. Even though the time can go up there, as of now in preparation phase, but it could be worth it. Understand the mistakes, right? So yes. Okay. If there was a recombinant cell options, ma'am, if there was a recombinant cell options, still the first would be transformant. Still the first would be transformant. Okay. Why? Because again, over here, both of them, Baba, recombinant one will also have ampicillin resistant. The transformant one will also have ampicillin resistant. The first thing before recombination is what? Before finding the recombinant cell is what? Finding the transformant cells, which is why the answer could be transformant cells. Okay? Clear? If clear, just give me a thumbs up. Everyone who feels this particular question is super clear to you, just give me a thumbs up. Ma'am. Yes, Sagar, tell me. Is this question clear? Perfect. Thank you. Let's go ahead. Question number 16. Identify the labels M and N in the following agarose gel electrophoresis representation. So those of you are not able to see it, I will be making it. This is M and this is N. So these are chotu things and these are going to be the bigger ones. So you have to find what is M and what are N over here. That is the question. So M is the smallest DNA bands and N is the largest DNA bands. Okay, and they clearly mentioned that the wells are over here. That means the DNA is put over here. Okay, now you have to tell the answer. What do you think? Okay. If question asked about insertional activation, then recombinant could be right. Yes, again depends on the question. Okay, Suchit says D, Shivanand says D, Sandy says A, my boob is also there, hello, it says D, Hina Kausa says D, Darshan says D, Fact D, Sai D, Sinchan says D, most of you, GS Suni, Sagar Chopra, Hassan, Vidya, Arif, Pradhani, GS World, Monica is also there, hello, Tasmia, Tanisha, Lilavati, Irana, hello. So most of you guys are saying the answer for this question is D. D is what? D means M is going to be the largest DNA bands and N is going to be the smallest DNA bands. Yes, I told you the N bands, the small bands which are there, they are going to move the farthest. So obviously the smaller ones are going to be found over here. This is a very easy question. The answer for this is D. If at all you get a question like this, oh my God, then probably perfect. Yes, hello Chetu, hello. Okay, perfect. Let's go ahead with the next question. Suppose DNA samples collected for DNA fingerprinting analysis are less than required quantity. Okay, so there's a criminal site, some crime has taken place over here, and the DNA sample that we have got is very little. Which of the following technique is helpful to make the sample sufficient for the analysis? I want to do the DNA fingerprinting and I need more sample. What is the technique that I will be using for it? Suchit says D. Deepti says D. Suchit, okay. Sanket says D. Sandy says D. Sagar says D. Shivanand D. User unavailable D. Okay. Hassan says D. Hina Kausar D. Anand D. Pooja Pujari. Suchit. Oh my God. Lilavati. Everyone. Deepti, Swami, Irana, GS Sunit, Anisha, Fax, GS World, Vidya, Mehboob, Sinchan. Note the kitty. D is what? PCR. Polymerase chain reaction. Always remember, this is the same type of question. You know what? You can get a same type of question from the chapter Biotechnology and its application as well. Because in that chapter, it is clearly been mentioned, if at all, the level of pathogen, listen to me carefully, the level of pathogen is really less. And I want to amplify, okay, I want to really find out that what is a pathogen in our body that is surviving in our body. Basically, we use PCR. PCR is going to amplify even the DNA of the pathogen out there, and we will be able to find out that as well. So PCR is the answer for this. Yes, polymerase chain reaction. Exactly, Shinchan. Yes, perfect. Next question. Identify the DNA sequence which can be cut using EcoR1. So what is the DNA sequence that can be cut using the EcoR1 enzyme? Ma'am, can you explain the DNA fingerprinting question asked in boards later? Asked in boards. What question was asked in boards? I have to think. Yes. What is the answer for this? Savita says it is D. Okay. Dhurandhar, oh my God. Dhurandhar says it is A. Suchit says it is B, dude. Okay. And Noor the kitty, cancer like easily fine. Okay. Pooja Pujari says C, and Soul says B. Arjun is there, hello, and says B. Sanket says B. Most of you guys are saying the answer for this question is what? B. Swami, I can't get it. This question you're not able to solve it, it's okay. I'll help you out. Okay. So the question is, identify the DNA sequence which can be cut using EcoR1. EcoR1, you know, all the enzymes, restriction enzyme which is there, all of them has a specific sequence. They are going to make a cut in only those particular sequence out there. What is the sequence for this? GAATTC. Okay, it is going to be GAATTC. Please remember that. GATC. Okay, Gayatri, take care. Whatever you want to remember, you remember. But please know, in the middle, the two letters which are there, they are doubled. AATT. Okay. Now, the direction is 5- to 3-. So, basically, EcoR1 will only find this particular region and make a cut. Make a cut where? It will make a cut between G and A. It will make a cut between G and A. Similarly, it's a double-stranded DNA, right? It will be a double-stranded DNA. If at all you make the complementary strand, C goes with G, T with A, T with A with T, A with T, G with C. Over here, what is the polarity? If here it is 5- to 3-, here it will be 5- to 3-. Where will it make a cut over here? It will make a cut over here. Okay, but the sequence is this. Now you have given this particular option, and most of you have given me the answer as what? Have given me the answer as B. Right? B. So let's go ahead with B. B is what? B is this. 5. A, G, 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 G. So listen, A, C, G, things is there. Listen, over here, G, A, A, T, T, C is there, right? G, A, A, T, T, C is clearly been spotted. It is from 5- to 3- direction. Yes. Now let's see if at all it is there in opposite direction also. G, A, A, T, T, C, it is there. It is there in 5- to 3-. That is why this will be the answer. It will be the answer. Okay? Please understand it. TC, remembering this sequence is important. Yes. Yes. Perfect. What does induced denaturation mean in PCR context? Please explain. Induced denaturation means it's not naturally happening. You're inducing it by providing heat and stuff like that. Cool? Clear, right? Everyone, it's clear. Just give me a thumbs up. Thank you so much. So B is the answer for this. And okay. Perfect. Next question. A student, while extracting DNA from Aspergillus fungus, requires dash enzyme to break open the cell wall. What do you think is the answer for this particular question? What do you think is the answer for this particular question? If at all I want to extract the DNA from Aspergillus fungus. Oh my God. D. Fax says D. Deepti says D. Dhurandhar says D. Hassan D. Sai D. Suchit D. Tasmiya, Savita, Hina, and Sandy, and Swami, and Vishnu. Vishnupriya, sorry, Monica, Arjun, Anand, all of you, most of you guys. Likhit is also there. Hello, Likhit. Okay, so most of you guys are saying the answer for this particular question is what? D. Why D, Baba? Listen, Aspergillus fungus is given. If at all you want to extract the DNA from a fungus, you know, the fungal cell wall which is there, it is made up of what? It is made up of chitin. So if you want to break open the chitin, you will require what? Chitinase. You will require chitinase, which is why D is going to be the answer for this particular question. Okay, done. Done. Perfect. Let's go ahead then with the next question out there, and it goes like this. Choose the bacterium which is not a source of rough endoplasmic reticulum. Rough endoplasmic reticulum. See. Okay, it is restriction endonuclease. Okay, restriction endonuclease. Choose the bacterium which is not a source of restriction endonuclease. Have you seen people zoning out in a live? Well, if you have not seen, that's me. That's RER, exactly, I know. Restriction endonuclease. Choose the bacterium which is not a source of REN. That is restriction endonuclease. Okay, C, Kavita says 20, okay. 20C, okay, fine. Sandy says A, Lilavati says A, Deepthi says C, Sagar Chopra C, Tanisha says D, okay. So there's a confusion between A, C is what I'm assuming, and also D, yeah. A lot of you children are saying A, some of you are saying D. Listen to me very carefully. Why did you say A? Haemophilus influenzae, H-I-N. You know Hind? You know Hind 2, Hind 3. That was the first enzyme that you got, the first restriction enzyme that you discovered. You got it from Haemophilus influenzae. So definitely that is not the answer. Okay, you get restriction endonuclease from there. EcoR1 you get from E. coli. Agrobacterium tumefaciens. It is not something, it's a bacteria, I do agree, but it is not something from where you get from where you get a restriction endonuclease. Okay, it's a vector that can be used for transferring things into a plant cell, but it does not, it's not a source of what? REN. Bacillus amyloliquefaciens, it might be, but not this. So C is your answer for this question. If at all you see another topic and you feel that, oh my god, this is not in your NCERT, so definitely this has to be the answer. You need to understand that, are all the terms in your NCERT being read properly first or not? Okay, so you think, okay, this, everything is there in the NCERT, so this will not be the answer. For in your NCERT, you might be pretty sure that Agrobacterium tumefaciens does not have any role in restriction endonuclease. It is therefore vector. So definitely that will not be the answer. Always remember that. Okay? Yes. So the answer for this question is C. Karthik, ma'am, I'm answering correct, but you're not taking my name. I'm so sorry, Karthik. Okay, I'm literally very sorry. Children, the thing is, the chat goes like this. Okay, and so basically it is difficult to read out all your names. It goes like this, right? Some might be repeated, so when it is getting repeated, I stop it right there itself. Right, so I'm so sorry, please forgive me. Okay, but I know I am missing a lot of your names, so yes. Tasmia, chemistry class is when? Day after tomorrow? Chemistry class is tomorrow evening. Yes, ma'am. Why this XP? Oh, do not know. Okay, clear. Let's go ahead with the next question then. Question number 21. By the way, from this question onwards, it's not PYQs, it's extra questions, newer questions. So try your best. Okay, Sanjana, good evening, dear. Okay, which of the following is not correctly matched sum for the organism and its cell wall degrading enzyme? You have to find which is not correctly matched. Algae methylase, fungi chitinase, bacteria lysozyme, plant cell cellulase. What do you think is the answer for this particular question? Very easy now, right? Very easy now. Everyone says the answer is A. That means that you have understood the assignment. Okay, that means that you are pro at it now. So fungi, it is made up of chitin, so chitinase is the enzyme to break it up. Bacteria, peptidoglycan, so you need lysozyme. Plant cell, you have cellulose, so you need cellulase. But algae and methylase do not go together, which is why this is the answer for the question. That is not the one that is correctly matched. Again and again asked, exactly. So I have kept this question, okay? This is not from your PYQ. I have kept this question because I saw many questions from this concept, right? Is it from NEET? No, this time I have not taken from NEET. I do not know whether these questions have come in NEET also. But yeah, this time I have not taken from NEET. Now, question number 22. Match the items in column 1 with their uses on column 2 and choose the right option. Okay, column 1 and column 2 we have to match. I always tell you one rule for match the following. Match A and C first. Okay, match A and basically whatever the option is last, match them first. Okay? Hey Cheta. Okay, so Escherichia coli, agrobacterium tumefaciens, and Thermus aquaticus is right over here, and you have to give me the answer. Here, my phone will have broken down. Okay. Duranda says the answer for this question is D. Okay. Kavita says D. Let me read out names. Okay. Hassan says D. Deepthi says D. Lilavati says D. And Sannidhi Shetty, hello. Fax says okay. Sanket says D. Sinchan Suchit has a different answer. Vidya says D. Hina Kosa says C. My boob says D. Vidya, okay. Vishnu Priya, Monica, and Kavana, Sai, Pradhani have taken the answer back. Tasmia, Sanjana, Chikappa is there. Hello, Chikappa. Okay. Noor the Kitty, D. Okay. Shardas, multiple answers. Karthik also says D. Sham Kamble also says D. Okay. Now listen to me. Irana, perfect. All of you doing pretty great. Okay. Lakshmi. Okay. So the answer for this question, since most of you have said D, let me match out that. E. coli, restriction endonuclease, thermostable DNA polymerase, and TI plasmid. E. coli is basically the one that will be going with this only. E. coli has EcoR1, that is a restriction endonuclease. Thermus aquaticus, which is there, you know that's a bacteria from where you take DNA polymerase, the thermostable DNA polymerase for PCR. Okay, so C goes with 2. Okay. C goes with 2. Now we have Agrobacterium tumefaciens. Agrobacterium tumefaciens has TI plasmid, which is why your answer for this question is actually going to be D, and you guys are absolutely correct, and I'm super proud of you. Sinchan also said D. Okay, got it. Sagar also said D. Okay, I'm sorry I missed it yet. Perfect. Okay, let's go ahead. Rafat, perfect, dear children. Okay, let's go ahead with the next question. If yes, just give me a quick thumbs up. Quick thumbs up. Fast. Where are you from, ma'am? I was born and brought up in Indore, Madhya Pradesh, but my roots are from Kerala. My parents are from Kerala. Okay, perfect. Let's go ahead with the next question out there. And it goes like this. Okay. Which of the following statements are correct? Now, whenever you see a big question, you have to be patient out there and read it properly, okay? There might be a lot of things to confuse you guys. You have to find what is correct over here. TI plasmid of Agrobacterium tumefaciens is used to deliver desirable gene into a plant cell. Is that correct? Is that particular statement correct? Check it out. Hint 2, always cuts the DNA molecules at a particular point by recognizing a specific sequence of 6 base pairs. Okay. Separated DNA fragments cannot be visualized without staining on an agarose gel electrophoresis. Okay. ORI is a sequence responsible for controlling the copy number of the linked gene. Linked DNA. Okay. And DNA is positively charged molecule. So you have these many options and you have to tell me what is the correct answer. B. Most of you says it is B. Okay. Good evening, Joseph. Okay. Most of you guys are saying the answer for this question is B, which is why I will not take much of your time. It is B only. It is going to be B. Why B? B says 1, 2, 3, 4 are correct. 1, 2, 3, 4 are correct, but fifth option is wrong. Why fifth is wrong, Baba? DNA is positively charged molecule. DNA might be like, oh my God, what is happening? DNA is negatively charged. It is not positively charged. It is negatively charged, which is why this particular statement is going to be wrong. What about other statement? It is absolutely correct. Agrobacterium tumefaciens has a TI plasmid, and that is used for transferring things into a plant cell only, not animal cell. So this is correct, a plant cell. Hint 2, it is mentioned in your NCERT. This is the first enzyme to be discovered, and basically it has a sequence of 6 base pairs. That is also correct. Separated DNA fragments cannot be visualized until and unless you put a strain. What is the name of that particular stain? Any idea? What is the name of that particular stain? DNA is negatively charged and acidic in nature. Exactly. Ma'am, 3 is wrong. Okay. Separated DNA fragments cannot be visualized without staining on agarose gel electrophoresis. That's correct. No, you cannot visualize it without a stain. Ethidium bromide, exactly. You need ethidium bromide, okay? So if at all you have separated the DNA and if you want to see it, you have to put the stain ethidium bromide. It is going to go into the DNA, and when you put UV light, it will be clearly visible to you. Okay, it comes out to be a bright orangish red band out there. Okay, now ORI site is a sequence responsible for controlling the copy number. I told you it is the one that is responsible for controlling the copy number. Then this particular statement is wrong, and I have given you the answer, which is why B will be the correct statement. Organic color bands, exactly. strain is ethylium bromide. You guys know that. You guys are awesome. Do you know that? If at all you didn't know that, you are. Okay, so perfect. Good. Is this question clear? Do you have doubts? If anyone has any doubt, please feel very free to ask me. Deepti, thank you so much. Yes. Sinchan, tell me, dear. Ma'am, reply, what? DNA fragments are also negatively charged? Yes, dear. DNA fragments are negatively charged and they are acidic also. Okay? Yes, perfect. Next question. Select the wrong statement. The presence of again, read it properly. Okay, again, read it properly. The presence of chromogenic substrate gives blue color colonies if the plasmid in the bacteria does not have an insert. Complicated. Okay. Retrovirus in animals have the ability to transform normal cells into cancerous cells. Do the retrovirus have that ability? You tell me. In microinjection, cells are bombarded with high-velocity microparticles of gold and tungsten coated with DNA. Since the DNA is hydrophilic molecule, it cannot pass through the cell membrane. What do you think is the answer for this particular question? You have been asked to find what is the wrong statement. And sometimes, I always tell you, they give you a lot of things to make you confused. Guys, be very sure about what you're answering. Check the options. You do not even have to think too much about it. It's very straightforward question. Yes, most of you guys are saying C. I'm seeing some people are saying B also. The answer for this question is going to be C. So whenever you get a complicated question and you read out the options and you feel that you know one option very clearly, it becomes all the more simple. You do not need to even dig deep into the other options out there. Here, C option. In microinjection, cells are bombarded with high-velocity microparticles of gold or tungsten. It is microinjection? That is biolistic method. That is beam gun method, not microinjection. And this is pretty much evident that it is wrong, right? So C is the answer. You have to find what is wrong over here. C is definitely going to be the answer. Got it? Always, whenever, I told you, so whenever you see a bigger question, you feel that, oh my God, what is there? I have to dig deep into that. I have to think of each of every, every process out there. I have to think about the method that is being used over there. Read out the question properly. You might get your answers there itself very easily. Okay, so yes. It's generally pistol. Oh my God, yes. Perfect. Got it? If yes, give me a thumbs up. My hippocampus is not hippocampusing. It's okay, it happens. Today is our day. Okay, thumbs up if it's clear. Perfect. Now, amplification, this is going to be the last question of the chapter, biotechnology and principles and processes. Okay, amplification of gene of interest by using DNA polymerase may go up to, if at all you are using this particular thing, okay, that is DNA polymerase, okay, for amplifying the gene of interest, it might go, amplification will go up to what? Any idea? 1 billion. Everyone has an idea. Okay, perfect. The answer is going to be what? 1 billion. It is going to be 1 billion times. Please remember this. This is important. 25th C. And with that, we have completed the first chapter for today's session, which was what? Biotechnology, principles and processes. And now we are going ahead with the next chapter, that is biotechnology and its applications. Do you have anything? One more, ma'am. One more. Durandar. What da? Conclusion. It's only biology which gives confidence. Other subjects. Is it? Thank you so much. Ma'am, the answer is already marked. I think no, while I gave for typing, it came out to be like this. Don't worry guys, I'll have some other methods to make sure that you do not give me the answers. By the class's end. No, the class is not ended. Okay. So children, I'll request you guys to take a 5 minutes break because your teacher also wants a 5 minutes break. Okay, so in 5 minutes I'll be back. I just have to find the AC remote. It's been too hot over here. And yes, so I'll have a break. And that break is going to be a Kit Kat break. You should not be eating Kit Kat. It's just an, in the ad of Kit Kat, they tell no, take a Kit Kat break. So it's just like that. Please, I'm not promoting any Kit Kats to be eaten. Okay, spare those Kit Kats and be right back by, if it is 6:18 now, let's be back by 6:23. Okay? Pappu, ma'am. Okay. So be back, and when you come back, make sure you call all of your friends who are confused in biotechnology in its application. Okay? Yes. Let's go. By the way, that was not a compliment. No, ma'am took it as a compliment. Are you aware? What happened? Oh, which gives now I get it. That was sarcasm. Okay, fine. Okay, guys, let's meet. Okay, at 6:23. Bye bye. Hello, 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 hello. Almost punctual. Yes, yes. Thank you, thank you, thank you. You know what? Your ma'am found the AC remote, but it's not working. Yes, which obviously is not important for you guys, which is why we will come back to what we are studying. Okay. So machas and machis, everyone over here, have you hit on the like button? If not, hit on the like button right now and hit on the subscribe button as well if at all you are new to the channel. Yes. So the thing is, the one which is used to tie my hair, that is also not visible. So your ma'am is, what do you call, someone who is, yeah, anyways. So guys, let's go ahead with the next question that we have over here, and the next chapter that we have over here, and that is biotechnology and its application. It's similar type of thing, so make sure you listen to me very carefully. And Tasmia, please do fast questions. I'll try my best to do so. The reason I am lagging behind is probably because I think some people, most of you guys answer the questions, but there might be some children out there who are not able to answer the question, and they do not feel to ask also, thinking that okay, lots of children are answering, so probably the problem is with me. I do not understand the concept, or I do not know it, and they might feel bad about it. That is the exact reason, even though when you give me the right answer, still I explain the question, because I feel that there might be some children like that. Okay, so yes, let's go ahead with the first question that we have, question number 26, and it goes like this. Read the statements and choose the correct option. Statement number 1, RNA interference takes place in all prokaryotic and eukaryotic organisms as a method of cellular defense. Okay, statement number 1 is that. Statement number 2, RNA interference is a pre-translational process. It is a pre-translational process. Now over here, what do you think is the answer? Statement 1 is incorrect, statement 2 is correct, and so on so forth. Okay, because I live so time is much going. Okay, sorry. Okay, so A is true. A is true. Sinchan says A is true. Hina says B is the answer. Vattikutti is there. Hello dear. Says it is B. Okay. Rekha says it is B. Sandy says B. Deepti says A. Sanjana says A. Tasmia says B. Okay. Hassan says 2 is correct. Okay, statement number 2 is correct. Okay, so my boob says B. Anmol Chopra is there saying B is the answer. Okay, myself Sagar. Okay, Sagar, hi. Ishwar Reddy says B is the answer. What do you think is the answer? Listen to me carefully. RNA interference takes place in all prokaryotic and eukaryotic organisms as a method of cellular defense. Wrong. Okay, it happens only in the case of eukaryotes, not prokaryotes. That will be wrong to say that. Okay, next thing, statement number 2, RNA interference is a pre-translational process. What do you mean by pre-translational? Listen to me. RNA interference as a whole means what? It means that you have an mRNA. Baba, just imagine this is an mRNA, and this mRNA has the superpower to make a protein. Right? It has the power to give information to make the protein. This protein can be toxic for the plant. This protein can be toxic for the plant. Who is giving the information? It is basically a worm that is known as Meloidigoin incognitia. Okay, so that has the mRNA. It is there for producing protein, and this protein can be toxic for the plant out there. Now, what is the process being involved over here when we say that mRNA is used to make protein? Translation is the process name. Okay, and we are going to mess around here itself. What are we doing such that the translation does not happen? We are going to give a complementary pair to this mRNA. This mRNA, which was a single until now, is mingle. Is not anymore single. And always understand, only a single stranded mRNA can undergo translation. A double stranded mRNA cannot undergo translation. So aren't we interfering over here? We are interfering over here. Don't you think it is happening before translation process? Okay, we are messing around the translation process, so it's happening before pre-translational process. Yes. Okay, it's not done after translation. It is done such that translation is avoided. So basically, it's a pre-translational process. This is going to be correct. Okay, so statement 2 is the only thing that is correct, and statement 1 is false, which is why answer is going to be A. It is going to be A. So everyone who basically did not give me the correct answer, I hope it's clear. Okay, Karthik, I am the girl, da. I am here for friendship. Okay, guys, over here, listen to me carefully. Come here to understand the concepts. That will be really good. Don't do loose talks over here. Okay. Clear? Ma'am, me also say option A, but you don't take Swami, I'm so sorry, dear. Okay. Is this clear? Is this clear? Okay. Perfect. Let's go ahead with the next question that is there, and that is question number 27. The human protein alpha-1 antitrypsin is obtained from. alpha-1 antitrypsin. Okay, this is a human protein, but you can make other organisms produce it. So where do you get it from? From transformed bacteria, from transgenic animal, from transgenic plant, or a plant from Western Ghats? Or a plant from Western Ghats. What do you think is the answer? Fatima Faiza is there. Hello, dear. Says B is the answer. Deepti says B. Sandy says B. Hassan says B. Shankar, hello, hello, hello. Baswara says B. Tasmia says B. Hina B. Anmol B. Chikappa says B. Okay. Rekha B. Always Sheikh. Hello, hello, hello. Says B. Okay. Fatima says cow. Okay. Okay. Cow is not the answer, by the way. Okay. And Tasmia says B. Kirtana says B. Sanjana says B. Good evening, Shankar. Omkar Andi. Omkar says B. Okay. Venkatesh say B. Shardas. Everyone. Vatikuti. And if at all I have left anyone's name, I'm please sorry. Please forgive me. Okay. So the answer for this particular thing is going to be transgenic animal. As simple. Okay. It is going to be transgenic animal. It is not transformed bacteria. It's not transgenic plant also. It is going to be transgenic animal. They have not mentioned in your NCERT whether it's a dog, cat or monkey or probably mice, anything. They have not mentioned it. So please understand, you just have to be worried about being in an animal, not a plant. Okay. Mubina, yes, you're also correct, champ. Okay, question number 28. Guys, was the other question clear to you? Everyone, do you think this question was clear? Sharon is also there. Hello, hello. Is this question clear to you? Let's go ahead with the next question. If yes, give me a quick thumbs up. It is used to treat emphysema. Exactly. You're going to get questions related to that. I'll show you, okay? Thank you so much, guys, for giving the thumbs up. Let's go ahead. Silencing of a specific mRNA translation could be achieved through. Now you have to think twice. Think twice before giving the answer. Silencing of a specific mRNA translation could be achieved through antisense RNA, RNA interference technique, both A and B, and microinjection. Okay, Sandy says the answer is D. Archana says it is C. Archana, hello dear. Baswaraj says B. Hina says B. Most of you guys are confused, say C and B. Okay. But I see now the number of children who are vouching for C, it is more. B, Baba. Swami says B is there. Okay, listen. The answer is C. I'm sorry. Listen. So over here, you know the complementary pair that we there. It's, for example, we say one RNA strand as sense. And the complementary strand for this particular RNA will be antisense. So over here, basically, we have mRNA strand. We need an antisense strand which is complementary to it to basically inhibit it. Yes, yes, ma'am. And this method is RNA interference technique. Yes, ma'am. So can I say both A and B and correct? Yes, ma'am. So the answer is going to be C. Microinjection is out of the league over here, so does not care. Okay. So the answer for this question would be C. Is this clear? Is this clear? If yes, give me thumbs up. Okay, sorry for the children who gave the wrong answer. Deepti. Okay? Done, done, done. I am Ganeshi. Yes, dear, I know you. Okay, perfect. Thank you for giving the thumbs up. Let's go ahead with the next question. Some strains of Bacillus thuringiensis produce proteins that kill insects. Which one of the following is not killed by proteins of Bacillus thuringiensis? Which is that particular thing that is not being broken down by, or basically not killed by, the proteins produced by Bacillus thuringiensis? D. Okay, Sandy says it is D. Okay, Archana says it is D. Deepti says it is D. Aarti Kulkarni is there. Hello. Says A. Okay. Shinchan says it is D. And Hassan says B. Hina says D. Kirtana says D. Okay, guys. Different answers. Most of you guys are now saying D is the answer. Leelavati, Swami, Sanjana, Shardas. So Tarun. Hello, hello, hello. Is D the answer? D is the answer. Tobacco bud worm, RV worm, cotton boll worms, they all come under what? They come under arthropods. Yeah. And they are the ones that are being killed with the help of Bacillus thuringiensis. But when I talk about tapeworm is basically chapta worm. Okay, it looks like this. It looks like a tape. It's a platyhelminthes. You know, flatworms? They are platyhelminthes. They are not coming under arthropods. There's a whole different file out there. Right? So which is why this is the odd one out. D is the answer for this particular question. Okay. 29D, ma'am, Pinne Karna, please tell us when is your next live. Actually, Malayali, that's so great. So guys, by the way, since someone has asked about this, I want to tell one very important thing. Tomorrow, I'll be coming live again at 9 a.m. in the morning. It can be 9:30 also, I'll tell you. It will be informed in the WhatsApp group. Please be there in the WhatsApp group. The chapter will be Microbes and Human Welfare and Organism and Population. Very important. Organism and Population is super important in terms of KCET. Make sure you attempt it. The reason that it is not, there is usually a gap of 1 day, is that on Good Friday I'll be on leave because I'm a Christian, I have to go to church. So yes, so please understand that if at all you want to attend the live, be there at morning 9 or 9:30. Okay, we'll be updated. Cool. Yes. Where is the WhatsApp group? WhatsApp group, Baba, if at all you close the chat, okay, and if you go to the description section of this particular video to get lecture notes and PDF, you will be seeing one particular link out there. Just click on that particular link. Okay, you will be moving ahead with the WhatsApp channel. Yeah, perfect. Okay, inshallah pinne kanam. Okay, bye bye. So let's go ahead. Okay, Baba, are you Malayali? Yes, I am a Malayali. Question number 30. A scientist wants to produce virus-free plants in tissue culture. Which part of the plant will he use as an explant? A scientist is there, he wants to produce a virus-free plant. Which part of the plant are you going to use? And over here you have the options: mature stem, axillary meristem, apical meristem, and mesophyll cell. What do you think is the answer for this? Okay, Leelavati says it is B, Deepthi says it is B, okay, Hassan says it is C, Sandy says C, Hina causes C, Fatima says it is B, Archana says, okay, Deepthi says it is B, Mahboob says it is B, Tasmia says B, okay, Prakash, hello Prakash, Suchit, Baswiraj, and okay, yes, always shake, everyone, most of you guys have different, okay, Irana, Kavana. Listen, the answer for this question is going to be B and C. That means it is going to be B. Over here, when you talk about a plant, always remember at the tip, what do you have? You have the apical meristem. Meristem means they are group of cells. Basically, it is made up of group of meristematic cells that can continuously divide. These cells which are continuously dividing, they are usually free of viruses. Suppose this particular plant, poor papa plant, is infected by the virus. But the apical meristem and also the axillary meristem, that means at this particular portion where you have a leaf over here, you have axillary bud. This particular bud also is made up of meristematic cells. So apical and axillary meristematic cells basically, or the meristem, are the area which are free of viruses. So you can use those particular portion for tissue culture. You can take that part and do the tissue culture, which is why the answer for this question is going to be B. Is there any doubt that you have? Is there any doubt that you have over here? Or is it clear? Okay. So, Karthik, I will be giving you one warning right now. Please do not talk amongst yourself, or the second step you all might know. Okay, so don't do that. And yes, Suchit, ma'am, when will you start first PU? Next week. Okay, let's go ahead with the next question. No doubts, since there are no doubts. Sagar, what is it on April 7th and 9th? Okay, perfect. 31st question. In mature insulin, which of the peptide is not present? In mature insulin, which of the peptide is not present? A peptide, B peptide, C peptide, and A and B peptide. What do you think is the answer for this? What do you think is the answer for this? C. Archana says the answer is C. Leelavati says C. Hassan says C. Tarun, okay, thank you. Sandy says C. Suchit says C. Anand says C. Nikita, hi, hello. Okay, Deepthi says C. Chikappa says C. Shurkant says B. Baswara says C. Prakash says C. Sanjana, Tasmia, Kannada Travel, Swami, Noor the Kitty, okay, always shake. Most of you, Ishwara Reddy, perfect, Ishwara Reddy, sorry, Kirtana, Evangel, hello Evangel, okay. Everyone are saying the answer for this question is C, which is why I lock up C. It is C peptide. Always remember, if at all you know the insulin, okay, you have A peptide, you have B peptide right over here, and in between that you have a C peptide. Okay, you have a C peptide. So basically, it's not like this. It is A here, it is going to be B here, and you have a C peptide right over here that is joining A and B. But the C peptides makes it a pro-insulin. That means it is not the mature form of insulin, it is the immature form of insulin. If you want to make it active, what do you need to do? You have to cut off. You have to cut off this particular chain from here. That is a C chain over here. What do we actually need? Basically, we just need A and B. We just need A and B in what? They have disulfide bonds, and this is what is found in the mature insulin. So in mature insulin, which of the peptide is not present? The answer would be C for this particular question. Clear? Okay, the Eli Lilly company, Humulin, exactly. Okay, works. Hello Ravi. Done. Guys, give me a thumbs up if at all it's clear. If at all it's clear, just give me a thumbs up. Hina, thank you, Sandy, Hina, and everyone out there. Okay, perfect. Let's go ahead with the next question. Why is it here? Oh shit. Okay. So guys, the next question apparently has to, the answer is already given over here. So please listen to me carefully. Okay. Which among the following is used to treat emphysema? The answer is already here, which is why I will not give the option for you to answer. It is human protein alpha-1 antitrypsin. We studied about it. Alpha-1 antitrypsin is a human protein that we get from a transgenic animal. What is it used to treat to? It is used to treat emphysema. It's a respiratory disorder, and please remember it's alpha-1 antitrypsin. It's alpha-1 antitrypsin. Okay? Works? And just a second. Now it's time to do something. Okay. Perfect. Let's go ahead then. Next question. 33rd question. Which of the following is based upon the principle of antigen-antibody interaction? Which among the following is based upon the principle of antigen-antibody interaction? Okay. Fatima says it is B. Always Sheikh says it is B. Shinchan says it is B. Kirtana says B. Hassan B. Anmol Chopra. What to do? Tasmia says Eliza is the answer for this question. Okay, B, that is B, right? Evangel, Suchit, and Deepti, Anand, Vijay, Kirtana, Shinchan, Chikuppa, Sandy, Ishwar Reddy, and Ramesh, and Basuraj, and Anmol Chopra, and Mehboob, everyone. Prakash, perfect. Sanjan also, perfect. Tanisha, everyone. Vattikutti, Irana, Noor the Kitty. So if at all I have missed anyone's name, I'm so sorry, okay? So answer for this particular question is going to be Eliza. Which of the following is based upon the principle of antigen-antibody interaction? I hope you guys know. Do not mug it up. Listen to me very carefully. Suppose there is Pappu. Okay, there is Pappu. And Pappu is worried whether he has HIV or not. Okay, that is the confusion that Pappu is having. So he went to give a blood test. What happens over here exactly is that, always remember, there is a chamber being made already over here where you have collected, you have the antibodies already prepared. These antibodies are what? These antibodies are something that are specific to HIV virus antigens. They are specific to HIV virus antigens. For example, from X person we know that who had HIV, they produce these type of antibodies against the HIV virus. So we have the antibody already over here. Now what do? We take the sample from Pappu and basically we check it out. We check it out and we get to know that the antigens are binding over here. But clearly, as of now, we are not able to identify it. Even now, we are not able to understand it. Until and unless we take the help of another antibody. This is known as secondary antibody. It will come and bind on this. When we put the secondary antibody, it will also come and bind on the antigen. And this antibody which is there, it is linked with an enzyme as well. It is linked with an enzyme. Now what do? You put a coloring substance over here. You put a substance over here. The enzyme acts on it. And if at all there's a color change and things like that, what is your understanding? That yes, this whole binding has taken place. That means antigen is there. That means Pappu could be infected with HIV. That is the logic of ELISA, enzyme-linked immunosorbent assay. Okay, this is important. Fine, this is important. Got it? Yes? Is it clear? They are disturbing, yes, I can clearly see that. Naveen, Maitri, as well as Ravi Barkar. Guys, please do not talk over here. Okay? Papu's mom's favorite, exactly. So yes. Is this clear to you? It's important. And on the other hand, it can be opposite also. Antigen can be kept over here. From the patient's body, you can take the antibody also. If at all the patient has been infected from the virus, obviously they will be having antibody as well. So always remember this thing. Miss ELISA, meaning ELISA stands for enzyme-linked immunosorbent assay. So over here, it is this antibody is linked with the enzyme, right? And it is going to show a color change when all this thing is going to happen. That is the meaning. Okay? Fine. Let's go ahead then with the next question that we have. Okay. And it goes like this. Question number 35. Cry1AC effectively controls what? Guys, over here you have the option. Root nematode, cotton bollworms, ringworm, corn borers. What do you think is the answer for this question? Ma'am, those type of question was asked in boards. Is it? Yes. Okay, guys, try Madi this particular question. Come on, boost up. Hassan says the answer is D, and Nagalinga says it is B, Sandy says it is B, okay, Swami says B, Baba, Sanjana says hi, Always Sheikh says B, Prakash says B, Sinchan says B, Pooja Pujari Vijay has a different answer, okay, Fatima says B, Deepthi says B, Ravi Barkar says, says B, Basraj says B, Kirtana says B, Sanjana says D, okay. Ma'am, T, Aita, Itla. Okay, listen, over here, study. Okay, most of you guys are saying it is B, but some of you are saying it is D as well. Why do you think that is the answer? The answer for this question, let's decode it. Always remember, you have cry 1 AC first and cry 2 AB over here. And basically, these genes are specific for cotton bollworms. Ma'am, how do you remember? Do you mug it up? No, there's a way to remember. First letter is C. First letter is C. Second words, first letter is going to be B. Okay, so ma'am, corn borer is also like that. What an information is this? So listen to me. Corn borers is a small name. Cotton bollworms is a bigger name. That is why you have two genes. Okay, you can remember like that. I know it's silly, but yes. Do you think all tricks are going to be so in what do you call something that comes out of a lot of intelligence? No. So yes. So over here you have one more thing, that is cry1AB. That is for what? That is for corn borers. So corn borers have just one particular gene. First letter is C and the next one is B. Okay, so the answer for this question is going to be cotton bollworms, since we are talking about cry1AC. Why it is not cry2? Listen to over here if at all you're confused. Cry2AC. CC. No, that is not possible at all. So because we want corn borers, right? So which is why answer is going to be there. Is this clear to you? Is this clear to you? AB, Amitabh Bachchan, 2. That's Abhishek Bachchan. Oh my God. Oh, yes. Clear. Is this clear to everyone? Yes, ma'am. Thank you so much. Let's go ahead with the next question. Now there, 36th question. Silencing of specific mRNA in RNA interference is what? In RNA interference is what? Karthik, you know what? We all love you, which is why we should not be here. Yes, let's go ahead. So silencing of specific mRNA in RNA interference is by, come on. Fatima says D, Deepthi says C, Kirtana says C, Irana says clear, thank you, Anand says D, Sandy says C, Deepthi says Chikappa D, Shurkan says okay, Prakash says B, Hina says D, Sanjana says D, perfect, Mahboob also says D, Nikita, Baswaraj, Shankar, Swami, Kirtana, Hassan, Irana, okay, Divyashri. Listen to me very carefully. Guys, those of you getting confused, the answer for this question is going to be double-stranded RNA. Why double-stranded RNA? So you have studied, ma'am, you only told in mRNA only one strand comes and attaches over here. So how can it be double-stranded? Listen, this was actually double-stranded RNA. And from this particular double-stranded RNA, one guy, only one, okay, one came over here and attached.
one came over here and attached. So even though a single strand is performing the function, but you actually got it from what? A double-stranded RNA. So which is why the answer for this question is going to be D. For those of you confused why it is not S as RNA, you need to understand, first it was double-stranded RNA, from here you got one strand. Okay, that is why. Who is the major thing? If it is not double-stranded, obviously it could not have survived. You needed to make sure that it survived, that is why you made it double-stranded, then you took one strand out of it. Okay, got it? I hope this is clear. Ma'am, please say whether agriculture quota is required for KCET. Yes, if at all you belong to that, yes, you can go ahead and give agriculture's quota. There is a practical exam as well, but you had to register, dear. You had to register for it. Okay. Question number 37. Silencing of a gene could be achieved through the use of. Silencing of the gene could be achieved through the use of what? Short interfering RNA, antisense RNA, both A and B, none of the above. You see how many times this question has repeated. RNA interference is one of the most important concepts out there. Do not miss it out. Okay. Vijay says yes, okay. Fatima says C is the answer. Sujit says C. Ramesh, what happened, macha? Mahbub says C. Kirtana says C. Rekha says C. Ravi says C. Hassan C. Vetikuti, Sandy, Baswaraj, Ravi, and Prakash, and Likhit. Pooja Pujari, Vijay, Sanjana, Sonika, hello Divyashri, Anand, always shake. Okay, so most of you guys are saying C is the answer. I think Sinchan also C. So both A and B, right? Short interfering RNA and antisense RNA. Short interfering RNA, ma'am, what is that? Listen to me carefully. Here it is written, it is used in RNA interference method, right? So basically we spoke about the double-stranded RNA, right? From this also, some specific part is being taken up, which you call as siRNA, short interfering RNA. And from this particular strand that we have got, one strand is being taken up for what? For being complementary to the mRNA. So basically, it's okay if at all siRNA or double-stranded RNA, anything is mentioned, it makes sense. So short interfering RNA, which is used in RNA interference, and also the antisense RNA. So in this short interfering RNA that is there, this double-stranded thing that is there, one strand comes and binds on the mRNA. One strand comes and binds on the mRNA, and that is going to be known as what? The antisense strand, which is why the answer for this question is going to be C. I'm always correct. Wow, perfect, Prakash. Okay. Yes, so C is the answer. If yes, if you have understood, can you just give me a thumbs up, children? Deepti, so my dear children, if at all you have not applied, I do not know what could be done as of now, because they had given so many options for you to go ahead and apply. I did the application form, so okay, let's go. Yes. 37th question is done, and now we go to 38th. Which of the following diagnostic tools allows the detection of very low concentration of bacterium or viruses by amplifying their nucleic acid? By amplifying their nucleic acid. So the thing is that the organism that we are trying to find out, whether it be bacteria or virus, we know that they are in the body, but their amount is very less. How do we find it out? Whether which organism is there? Which is the process for that? ELISA, PCR, autoradiography, or rDNA technology? What do you think is the answer for that? Suchit says B, Hina Kausa says B, and Fatima says B, Rekha, Hassan, Likhit, Sandy, Shankar, Kirtana, Fatima, as well as Always Sheikh, Supreet, Ravi Barkar, Godzo, perfect, Nagalinga, Sanjana, and Deepti, Mahbub, Kirtana, Nikita, Vijay, Pooja, everyone, Irana, Evangel, Shinchan, Prakash. Okay, I think I have taken Vatikuti, everyone doing absolutely great. Pavan, Sudarshan, Anmol, Chopra. Okay, so the answer for this question is going to be B. We discussed about it. It's PCR. PCR is basically what? Polymerase chain reaction, where what you're doing, even with little amount of thing, you can amplify it and you'll get to know which organism it belongs to, right? So it's an easy question. We have discussed about this in the other chapter as well. Before that, I want to, ma'am, please do inform when I can apply for that. Deepti, I'm not sure whether you can apply for that now, because the time period has been officially over. The editing application window is officially being done. That's the reason I'm like a little confused, okay? Okay, now listen to me very carefully, because this live might end up anyway. So Anmol said that I was not reading out the name. Listen, understand me, okay? I am a teacher. I'm over here explaining things to you. You are just answering the question, right? You are answering the question. You are thinking. You are answering the question. I'm a teacher. As a teacher, I have to explain this. Then I come back to you. I try to engage really well with you guys. That's why I know your names, right? But obviously, at times, I could miss your names here and there. And if at all I keep on reading the names, what is going to happen? The time it is going to take up a huge lot of time. I do agree that you feel demotivated if at all I do not take out your names, but sometimes we have to be understanding, right? It's not like I never take your name, right? So please understand it. It's very important. Your understanding, if at all my understanding is important, is equal just as yours. So yeah. Okay, let's go ahead then with the next question that we have over here, and that is this. Question number 39. Some multinational companies have exploited the traditional knowledge of the indigenous people to produce commercially important bioproducts without their consent. This is an example for example, you saw the example for basmati, okay, basmati rice which is there. An American company said that, hey, Macha, it's mine, you do not produce it now. How can that be possible? That's not fair, right? Indian farmers are producing it, and without their consent, you have patented it. You have said that, oh, no, you cannot do it, okay, you cannot do anything out there. This is ours now. So what do you call this particular thing as? Most of you guys say C is the correct answer for this particular question. Yes, Chandana, Rekha, Sanchan, everyone. So C is the correct answer for this question. Is it C? Is it C? Is it biopiracy? Yes. Some multinational companies have exploited the traditional knowledge of indigenous people to produce commercially important bioproducts without their consent. This particular thing is an example of biopiracy. Okay, what does this company do? They put patency. And this particular process where you're not taking the consent, it is known as biopiracy. So the answer for this particular question is C. Sometimes children get confused with this and this. Do not get confused. You have the patent, okay, you can legally do it, but here you're not doing it legally. You're doing it without their consent. This process is known as biopiracy. Okay. Anmol is totally fine, Macha. Okay. I hope it's crystal clear to everyone out there. Okay, let's go ahead with the next question. If it's clear, can you give me a thumbs up, children? Likhit, thank you so much, Da. Ramesh, ma'am, for BSc Agriculture, when should I want to apply? The application window has closed, right? Okay. Perfect. Let's go ahead. So question number 40. Which of these is not an advantage in genetically modified crop? Which of these is not an advantage is being asked. Not an advantage. Increases efficiency of material usage in plants. Reduces the reliance on chemical pesticides. Enhances the nutritional value of the food. Increases the post-harvest losses. What do you think is the answer for this? Hina Kosa says A. Fatima says D is the answer. Fact says A. Guys, read the option carefully. Sandy says D. Vaikuti also says it is D. Rekha says B. Sanjana says it is Sanjana, thank you. Vijay says D. Shinchan says D. Anand says D. Okay. Fact says D. Ravi Barka D. My boob D. Chikopa have a different answer. Hina D. Always Sheikh and Kirtana and Vaikuti, Shinchan, Deepthi and Lavender, Sudarshan, Evangel, Baswaraj, Vijay, Likhit, Anmol, Maha Shri, okay, okay, so sorry, okay, so Sonika, everyone, so all of you who gave me the correct answer, listen to me carefully. The answer is actually D itself. This is the incorrect statement, not an advantage, right? Increases the post-harvest losses. How can that be an advantage? Suppose I am a farmer, okay, I did harvesting and everything, and then I kept all my things under one particular roof thinking that it will be safe. Now over there, some bugs and all things came and ate it up. What is happening? This is a loss for me, okay? When an organism, basically when a plant is genetically modified, it's made sure that it reduces the losses, not increases the losses. So over here it is written that increases the post-harvest losses. So obviously this is going to be wrong. Increases the efficiency of material usage in plants? Yes, it does. What do you mean by that? Sometimes it feels to be negative. Listen, I have a plant over here. Okay, this is one plant and this is another plant. This plant's name is Chutku plant. This plant's name is Butku plant. Listen to me. Chutku says that I need magnesium, I need phosphorus, I need nitrogen, I need potassium, then only I'll survive, okay? And I need it in 50 grams, 550 grams and 60 grams and things like that. Then only this particular plant is surviving. Whereas Butku says that I can survive even if I have a little amount of phosphorus, little amount of nitrogen, little amount of this thing. I can survive nicely. what is good about Budku over here? That is basically it is able to survive with little amount of minerals. Its efficiency of mineral usage is nice. It's good. It's better. With the little amount of mineral, it is able to survive itself, right? So increases the efficiency of material usage in plants is a good thing. It's a good thing. Over here, it needs a lot of things and it might not be available. So obviously not good. First, survival. Reduces the reliance on chemical pesticides. Yes. So over here, suppose Budku is the one that is having some Bt cotton gene and stuff like that. What is happening? Basically, it can rely even if there is an attack by any sort of insects, right? Because obviously you do not need chemical pesticide. Whereas over Chutku, I have to put pesticides over here that can go mix with the soil, and that will be really bad. Enhances the nutritional value of the food. You might have seen, you might have heard about Africa, right? So over here, in the case of they faced severe malnutrition and stuff like that, and there were a lot of problems around it, which is why people came up with the new thing that is, the staple food. Most of us eat either chapati or rice, right? That is how it goes. So why not make the rice good, have a better nutritional value apart from the carb which is there? So with the help of genetic engineering, they came up with one very important thing, that is they infused. They made sure that the amount of vitamin A in the rice is increased. The amount of vitamin A is basically increased, which is why the color of the rice, it came out to be golden, which is why you call it golden rice as well. So people who had it basically had a spike in vitamin A, which increased their, the eyesight and stuff like that. Okay, Budku means dwarf. Yes, I know. Get it? So please remember, enhanced nutritional value of food. Yes, that is also a good feature of genetically modified plants. So your only answer for this question is D, and the reason why I explained the other option is that you might be able to spot some other questions based on this. Okay? I'm scared about results. Listen, you gave the examination. Can you do anything more about it right now? No. It's not in our hands, is it? No. But worrying about it, being in a bad mood, is that in our hands? Definitely yes. Okay. So you obviously have a right to be sad. Okay, that should be there. You should be understanding. You should be going through all the emotional ups and downs are there. That is how we human beings are. We should feel it, embrace it as well. But that should not sulk us down. So it's okay. We have given the examination. Let's face it anyways. Okay. Fine. Question number 41. How many questions are there? 50 questions are there. So 9 more. Okay, choose the correct set of, just a second, okay? I think, no. 5 more questions are there, only 5 more questions. Okay, not 50 questions are there, only 5 more. Cool? Okay, choose the correct set of statements regarding golden rice. It's a transgenic variety of Oryza sativa. Second thing, it contains a good quality of vitamin A. Third thing, it is a principal source of vitamin B. Fourth thing, the grains of the rice are golden in color, so the rice is commonly called golden rice. What do you think is the answer for this particular question? I know the first statement is not given in your NCERT, but yes, you still can find the answer for this. Sanjana and Chetu, dear, don't. Okay? Sudarshan says the answer is C. Fatima says it is B, is it? Khiladi Gamer says hi dear. Okay, Sudarshan says C, Hassan says D is the answer. Okay, Sandy says D, Fatima says D, Likhit says D, Kirtana says B, Anmol says A, Vaitikuti says D, okay, Lavender says D, Likhit says D, Sanjana C, Shinchan says D, Pooja Pujari C, okay, Ravi Barkar D. Most of you have given me the answer. Hina Kausa D, Madhu, Sudan A, Ramesh, Kirtana, Baswaraj, Vaishalim and Mr. Shrikant, and Chetan, Chetu, Badrinath, Sudekha, Deepti, Anmol, everyone, Evangel. I hope I'm not missing out on any names. If I am, please sorry. So most of you guys said the answer for this question is D, is what I'm assuming, right? D is what you're saying. Let's see what is D. 1, 2 and 4 is correct. Is that so? It is transgenic variety of Oryza sativa. Oryza sativa is scientific name of rice. So it is definitely a transgenic variety. You are making changes, that is why transgenic. It contains a good quality of vitamin A. We just discussed about this. It does not have vitamin B, so wrong. Okay, the grains of the rice are golden. Why? And that is why it is known as golden rice. Yes, so the answer for this question is going to be D. And don't you think sparing some time there was helpful for you over here? That is how it goes. Okay, so the answer for this question is D. For everyone who gave me the wrong answer, I hope you have got the correct answer now. You have understood it now. If yes, give me a quick thumbs up. If yes, give me a quick thumbs up. Ma'am, when is the second PUC results? Could be in the second week of April. Yes. Thank you so much, and let's go ahead with the next question. Question number 42. Identify the correct labels for A, B, C, D marked in the figure given below. So over here, we have A, we have B, and we have C. Are you able to see it? If not, I'll be showing it to you. Okay. This is what they represent as A. Okay, this is A. This is mentioned as B, and this is mentioned as C, and this particular strand is mentioned as D. Okay, just have an understanding of it, and I'll show you the options. The options, they go like this. A is pro-insulin. Okay, so I can write it enlarged. This is A, this is B, this is C, and this one is D. Yes, now you can give me the answer. 41st question, please, again, once more. Please explain once more. I'll do that, Pooja Pujari. Okay, just wait for some time. I'll definitely make sure that you understand that. Arfat. Oh, okay. Likit says D is the answer. Okay. Mahbub says A is the answer. Vaishalim says it is D. Okay. Kirtana says it is D, and Sanjana, it is D. Lavender says D. Anmol Chopra says B. Sandy says D. Okay. Basuraj says D is the answer. Anand says D. Vijay says D. Sudarshan, Ravi, Rekha, Rafat, and Manasa, and Charan, and Chetu, Pooja Pujari, Kavana, Hina Kausar, Hassan. Ma'am, the question is out of my mind. It's easy. Try to solve it. Okay. Deepti, Shrikant, everyone, perfectly going good. Shankar Naik. So listen to me, the answer for this question is going to be this whole thing. They have not mentioned any particular strand over here. So A, if we have to choose, it can be A peptide, or basically it's about the whole thing. So it can be A peptide, but that is not an option. Only option that is given is pro-insulin. So basically we assume that this is pro-insulin. Okay. Now we want to form the mature form of insulin. I told you in mature form of insulin, what is there? A peptide and B peptide will be joined. What about the C peptide? That is being removed. So over here, C peptide will be a separate thing that is out there. So what is B over here? B is the A peptide. B is the A peptide. What is C over here? C is going to be the B peptide. It is going to be the B peptide. What is D over here? D is going to be the free C peptide. It is the free C peptide which is out there, which is why your answer for this question will be this. Okay, it is going to be this. This is the A, B, C, D option. It is going to be B. Cool. Is this clear? D is free C peptide. Guys, will you stop talking in between? That's so sad. So D is the free C peptide that is up there. Is it clear to everyone? If yes, just give me a quick thumbs up. Please explain once again. Definitely I can. For sure. Listen to me carefully. Over here, you have A peptide, you have B peptide, right? Fine. And they both are joined actually with the help of some disulfide bonds out there. Okay. Now, there is a bond up over here, and this is what? The C peptide. This is basically C peptide. My cough. Guys, just give me a second. Okay. Yeah, kar diya. Okay. So over here you have A and B peptide. In between that you have C peptide, which makes it what? It makes it the pro-insulin. That means it is not mature. It is going to be immature form of insulin. It's not in the working condition. Now to make it work, what do we have to do? We have to remove the C peptide, right? We have to remove the C peptide chain from here. So we remove that. When we remove it, what do we get? We get the A and the B chains. The A and the B chains will be still attached to each other with the help of what? Disulfide bonds. What What is left behind? Papa guy, that is the C peptide. This is a free C peptide that is staying out there. And that is exactly the question. What is this particular structure? That is going to be proinsulin. What is B over here? It is marked as B, but please understand it is A peptide chain, right? What is C over here? The C is marked as this particular chain, so it is B peptide. And finally, what is this particular thing, last thing that is left behind? It is going to be the C chain, which is why the answer for this question is going to be B. Okay? Yes? Fine. So, next question. Question number 43. And it's okay, it happens. Ravi Barkar and Shurkant and, Sanjana. Guys, please do not talk amongst each other. Please. It will be really great if at all you do not. Okay? Please understand my situation. Yeah? Question number 43. Which of the following statements about transgenic animals is or are false? You have to find which one is false out there, right? So, transgenic animals are designed to study how genes are regulated. Are they? Are they designed to study how genes are regulated? Second thing, they are specially made to serve as models for human diseases. You have heard about it. Cancer, Alzheimer's, cystic fibrosis, everything could be understood in a better manner with the help of transgenic animals. Transgenic cow Rosie was created to produce the human protein alpha antitrypsin. Okay. Fourth one, transgenic mice are used to test the safety of the vaccine. You know, vaccine safety testing is done on these particular organisms out there. What do you think is the answer for this particular question? And please understand, you have to find which is wrong, which is false. Okay, C. Most of you guys are saying C is the answer. What is C over here? C is 2 only. 2 is wrong. They are specially made to serve as models for human diseases. Oh, sorry. Yeah. Is your answer? Think about it. Think about it, guys. Think about it. We're getting things wrong. Is C the answer? From here, you have to choose the option A, B, C, D. Okay, so now let me read out the options. So Madhusudan says A, okay, Vatikuti says C, Likit says C. I do not know if it is the other question or this question only. Now everyone is saying A is the answer. Macha, what is the answer for this particular question? It is going to be A. Why A? Listen, we have to be very careful with this particular thing. 3 is the only thing that is incorrect. Transgenic cow Rosie was created to produce the human protein alpha-1 antitrypsin. Wrong. Alpha-lactalbumin. With the cow, you produce milk, and the milk has the human protein alpha-lactalbumin, not alpha-antitrypsin, which is why this is wrong. Papa cow is like, I do not know this happened, so this is wrong. Okay, now I hope it is clear. Clear, Macha? Divyashri, it is going to be tomorrow morning. Everyone, is it clear? Perfect. Perfect. Thank you. Now, 44th question out there, and it goes like this. When a person with defective adenosine deaminase was treated, which of the following steps are performed for gene therapy? So if at all a person is not having the gene for adenosine deaminase, and basically they're treated with the help of gene therapy, what are the steps that are being followed? It goes like this. Lymphocytes were extracted from the bone marrow of the patient. Lymphocytes were grown in a culture outside the body. Erythrocytes were transformed with the normal ADA genes. A functional ADA cDNA gene is introduced into the lymphocytes using a retroviral vector. What do you think is the answer for the question? And make sure you read the options very carefully. Okay? Clear, Machi. Thank you so much. Why not? Okay, why not 2? You're talking about this? Because they are specially made to serve as models for human diseases. It's correct. It's not false statement. You know, for understanding cancer, cystic fibrosis and all, you use animals. You see what are the changes that happens in the animals when they have got cancer. And basically, when you understand the changes, basically you can help in finding the cure as well. Right? Yes. Okay. So Mehboob says it is B, Sandy says it is A, Sanket says B, and Sandy says it is A, Rupashri A, Likit says B, Fatima says A, Mansa says B. What is this? A, B, C, D option for Mia. Guys, come on. Shankar says B, okay. Vatikuti says D, Ravi Barkar, Shakan says B, okay. Vaishalim says A. Parvati says A, Machi. Okay, perfect. Okay. So guys, listen to me carefully. The answer for this particular question is going to be, let's decode it. Lymphocytes were extracted from the bone marrow of the patients. Do we do that? Yes. We take the bone from the patient. Suppose Pappu again. Okay, Pappu is having some problem in his lymphocytes. In his lymphocytes, the gene is not there. That is ADA gene is not there. That means toxic substances are going to build up. So what do we do first? We take the lymphocytes from outside. From his body, we'll take it outside. We are going to culture it. We are going to culture it. After culturing, what do we do? Basically, with the help of a retrovirus. You know, retrovirus are the ones usually that affects us. So now we are not going to let them affect us. Rather, we are going to put our gene of interest in them. And basically, when they infect us, they are going to pass the gene of interest to us. Okay, so over here, I cultured it. Then what did I do? Into my cells over here, the lymphocytes over here, I inserted the gene of interest, which is known as cDNA, the complementary DNA. I put it with the help of what? With the help of retrovirus. Now everything is sorted. I have that particular gene in all of my cells. What do? Again, I take the cells and put it back into Pappu. That is the logic out there. So lymphocytes were extracted from the bone marrow of the patient. Correct statement. Lymphocytes were grown in the culture outside the body. Correct statement. Erythrocytes were transferred with the normal ADA gene. Why erythrocyte? We're talking about lymphocytes, no macha. Why erythrocytes? So over here, this particular thing is going to be wrong. Okay, functional ADA cDNA is introduced into lymphocytes using a retroviral vector. You saw with the help of a retroviral vector, you actually inserted a cDNA into the cells. So this particular statement is going to be correct, right? So what can we say? 1, 2 and 4 are correct. 1, 2 and 4 option where is there? 1, 2 and 4 option is not there. Wait. Dun ta na 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 dun ta na 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 na. 1. Did I do something wrong? Or are the options wrong? Okay. There's some problem. D, Machi. Guys, let's keep this particular question on hold. I do not understand. Since we're talking about lymphocytes, it should be lymphocytes itself. Okay, anyways, let's keep it on hold. Okay. Ma'am, bone marrow has RBC2. Yeah, I do agree that we can take RBCs as well. We can take erythrocytes as well for doing the whole process. But here, since they're talking about lymphocytes only, that is a concerning factor. Because talking about erythrocyte is not a wrong thing. But when you're talking about lymphocytes as a whole and in between that erythrocyte, but yes. Anyways, let's keep it on hold. Okay, let's keep it on hold. I think there is some problem. This is, as I said, it's not a PYQ question. I took it from this is like for preparation, it's an extra question that is there, so we can keep it on hold. Okay, yes, tomorrow I'll come and in the morning lecture we will be solving this question first. Cool. Yes, next question that we have over here would be this one, and please remember this is the last question for today's session. And the milk produced by Rosie, a transgenic cow, is known to be rich in which of the following? In which of the following? The milk produced by Rosie, a transgenic cow, is known to be rich in which of the following? Sugar, protein, fat, or vitamin? Sugar, protein, fat, or vitamin? What do you think is the answer for this? So many questions, so many answers, and now I have to read it up also. Why? Because this is the last question, right? Charan Kumar says B, Deepthi says B, okay, Vetikuti says B, Anmol says B, Soul Mani says B, Hassan says B, Madhusudan D, Hina Kaushal says B, Anand says D, okay, Kirtana says B, Mrityunjay says C is the answer, Baswaraj says it is B, and Fatima says B, Likit B, Deepthi says protein, okay, Rekha says D, Madhusudan, Vijay, Sutagati, and Sanket, Vaishalim, Mahboob, Soul, Mani, everyone. Mr. Shrikant, Nikita, Pooja Pujari, Baswaraj, I hope I am not missing out anyone's name. Darshan, Divyashri, listen to me carefully. Hanamata, Metri, okay. So everyone, Arvind. The answer for this particular question, Rupashri and Anmol Chopra, the answer for this particular question is going to be protein. We know that with the help of the cow, Rosie, we were producing milk, and this milk was rich in human protein, alpha-lactalbumin. It was a human protein, alpha-lactalbumin. Do not go and write alpha-1 antitrypsin. That is for treating emphysema. This is human protein, alpha-lactalbumin. Very, very important thing. It's a protein, not a fat, a vitamin, or a sugar. Cool? Now, so yes, with that, what do we understand? We understand that the live is officially over. And one very important thing that is, matcha means, it's like, it's a Tamil word. Sometimes we talk in Malayalam also like that. Matcha means like a friendly sort of thing, like, hey, you send a dude and some, I do not know how to explain that. But yeah, it's a friendly thing that you call to someone who is probably of your vibe or your age or things like that. Okay, but obviously you are not of the same age. But yes, I feel that I am like you. Okay, I don't feel that I'm too old enough, so I want to be your friend, which is exactly why I call you Machi. Okay, so yes, ma'am, when is your next class? So listen to me very carefully. My next class is going to happen tomorrow at 9 or 9:30 a.m. I'll just update it in the group. Most probably 9 a.m. itself. And please make sure you join the WhatsApp group, because there is where all the updates are going to come. Okay, and it is going to be on the chapter Microbes and Human Welfare and also Organisms and Population. Everyone needs to be there. Listen, I have seen so many children have been very consistent with the studies. From the first lecture onwards, you are here. With that, you get an understanding that we are covering PU2. And you should feel very proud of yourself, because I am definitely proud of everyone out there. And I'm literally sorry if at all I did not take your name or things like that. It was a rush thing. That is exactly why I was not able to. I'm really sorry for that, but I appreciate your efforts. You guys are awesome, and I really, really wish you get into the best college, your dream college out there. Yes? And anyone who has a doubt over here can stay back and ask me the doubt. Rest everyone who do not have a doubt can obviously leave the class if you want to, yeah? Likit, thank you, ma'am. Love you too, dear. And Sureka, thank you. Ma'am, KCET Marathon, we'll be doing it towards the, towards the examination, yeah? Give me a WhatsApp group. So it's in the description section of this particular video. If you close the live chat, you will be able to find the description section. Click on that, and there you will be having. Evangel, you're most welcome. So tomorrow everyone will be there, right? Yes. Thank you, Samad. Love you too, everyone. Vijay, and master stroke, then PYQ. Ma'am, master stroke, then PYQ. Yes. So master stroke for PU2 chapters are already given. For PU1 chapters it's left. I do agree. But yeah. Okay. So tomorrow morning, be there. I'll be very much excited. Tomorrow is Hanuman's birthday, is it? Okay. Man, app is not opening. That's not the case. It will be opening. Try it out. Reload once more. It is helpful for NEET or not? Listen, for NEET you have to practice NEET specific PYQs. This can be just a little help, but you need to focus on NEET PYQs. Okay? Okay, so yes, Paswaraj, thank you, friend ma'am. Oh, that's so sweet. You're most welcome. Okay. And yeah, guys, then let's end up this particular live. If I don't see any doubts, someone was asking me some question. 41st question repeat. Who was that? Vaishalim, thank you, love you man, love you too dear. Parvati, thank you, love you too, bye bye. Is there any doubt that someone was asking me the doubt? If you have that, please answer that right now. I mean, question me right now, I'll give you the answer for that. Thank you, Vidya. See you soon in the morning. Thank you, Shrikant. It means a lot. Okay, so I think there is no doubt, and which is why we'll go. Okay, so see you soon. Good night. Have a nice sleep. Don't let the mosquitoes bite you. Bye bye. Chetu, bye bye. Tomorrow class time is, I'll write it up. Okay, also one very important thing, those of you who have not joined KC30, please understand it's a crash course, KC30 crash course, and there is a 500 rupees off that is ongoing for KC30 crash course, where you get PU1 and PU2. There will be full syllabus coverage, there will be really practice papers given to you, there will be daily live sessions also. It has started from 21st of March, so if at all you're interested, make sure you take it quick. It's very important, okay? So do that. Yeah, and there is ABHYAS KCET test series for those of you who are just focusing on question practice, and that is also important. It's just available for rupees 999. So check out if it is of use for you, and that's all. Wishing you the best. See you really soon. Bye bye.